Good morning, dear delegates. I welcome you to MicroCon 2024, the 47th Annual Congress of the Indian Association of Medical Microbiologists, organized by the Department of Microbiology, BJ Government Medical College, Pune, in the cultural capital of Maharashtra. We are having a host of pre-conference CME today, and we are delighted to have you join us for one of the most remarkable lineups of 18 pre-conference workshops at MicroCon 2024. Our pre-conference workshop at serial number 10, titled Introduction to Targeted Next Generation Sequencing in MDR-TB Management using Oxford Nanopore Technology, Ampore TB, is organized by Department of Microbiology, BJ Government Medical College, Pune, and is coordinated by Dr Rashmita, Assistant Professor from my department. The faculty will provide you with practical skills and knowledge in application of nanopore sequencing for the rapid and targeted detection of multi-drug resistant tuberculosis, that is MDR-TB, following WHO recommendations. The workshop will emphasize the use of nanopore sequencing technology for real-time genomic surveillance, enabling prompt identification of resistance markers and guiding proper treatment strategies to combat MDR-TB. We encourage you to actually take part and understand the intricacies of this vast technology that is bound to transform the future of modern medicine. I welcome you again.
Most advanced workshop which we have arranged here. We have arranged 18 workshops, pre-conference workshops in our conference, MicroCon 2024. I think you must be aware, we have bombarded you with messages, so that it is very clear to you. But I am very happy for all of you that you have landed in the best of the workshops. Because this is a test which is going to be launched. It is going to be commercialized very soon, and WHO has almost, I think it has given its accent to it.
I am Dr Rashmita, the coordinator for the workshop, and I have with me my team members, Dr Rashmita and Dr Nyabo from Jeejeebhoy, Pune, and our extended team from Oxford Nanopore Technology. So we have to start with Dr Narendra Singh, Rebecca Smith, and Kristen Scott would be our expert for the day. So as we introduce us, I'm sorry, So as we move forward, I'd like all of you to introduce yourself to the room. So can we have the mic?
Institute of Medical Sciences, Sevadar.
A very good morning to all of you. I am presently working as an associate professor in the Department of Microbiology, Byramjee, and my name is Dr. Rashmita.
Good morning, all in law. I am Dr. Maheshwari, working as associate professor in the Department of Microbiology. I am from SR Medical College from Chennai.
Good morning all. Myself Dr. Deepika. I am first year postgraduate in microbiology, and I am from the Government Medical College.
Good morning everyone. I am Dr. Sangeeta, Professor of Microbiology from Rajajeshwari Medical College, Bangalore.
Hello everyone, good morning. My name is Nick Chia. I'm the Vice President for Medical Affairs from Byramjee, and I'm based in Singapore. So look forward to learning from each of you today. Thank you.
Hello everyone, my name is Prerna Sadhi. I'm working as a research fellow in the Department of Microbiology at British County Hospital.
Good morning, I am Dr. Vidya Nirukar. I head infectious diagnostics, autoimmunity and allergy, and toilet diagnostics.
Hi, good morning everyone. I'm Tushar Pune, India Marketing India South Asia
Hello, very good morning to everyone. I am Department of Microbiology and the National Application Support for the Molecular Biology Division, looking after the South Asia region for Byramjee.
Hello, good morning everyone. Myself Swapnil Dome, working as an application specialist for Byramjee. Thank you.
Good morning everyone. So this is Sachin here. I look after the marketing for Byramjee for molecular divisions. Thank you.
I thank you all once again for introducing yourself. I'd also like to congratulate each one of you in this room to be a part of this workshop, as you are among the first batch of participants in India to be introduced to this groundbreaking solution that Oxford Nanopore Technology has come up with. If I am not wrong, this is the first batch in India who would be knowing this solution. So a big congratulations to each one of you. So we'll start the session. I request Rebecca to please come up. So Rebecca has joined Oxford Nanopore Technology as a field application scientist in September 2022, and she then moved. Okay, sorry. I'm sorry. Can I have the agenda? Oxford Nanopore Technology and the Ampore TB lead clinical at Oxford Nanopore Technologies, is a dedicated and a passionate contributor to global fight against the tuberculosis with high burden regions and limited resource settings. His expertise spans over 15 years in global health and international business development. And he is currently the global access lead for clinical at Oxford Nanopore Technology, where he is focusing on expanding access to MDR and XDR-TB diagnostics through strategic global partnerships. So, questions?
Okay, so good morning, everyone. Thank you very much for the warm welcome and warm introduction. We are very pleased to be here and really honored to be in the first session for Ampore TB in India, to be really present here, and thank you for the invitation. So, as Dr Rashmita said, my name is Christian Konsom. I'm the Global Health Lead for Oxford Nanopore Clinical, which means that Oxford Nanopore Technologies has a small team of clinically, let's say, people who are focusing on the clinical solutions and how to implement the technology for clinical solutions. You know, because until now we are very well established in the life science research tools. So with this, we have developed the Ampore TB test, and I will give you a very high-level overview of the test, and then I will let my colleagues Rebecca and Lanuk to really dive into the technology and explain the technology, how it works, have also hands-on time, and really explain to you how accessible it is. And I'm also pleased to welcome our partners for global distribution of Ampore TB, and I will explain in the slides. So partners from the company Byramjee, who presented themselves already. So we are really happy for this partnership. And I will explain a little high-level overview how this partnership works and how we are approaching it. So further ado, I will dive into the presentation. So I will focusing on three kind of high-level points, which is the WHO guidelines, which were mentioned already, the Ampore TB test itself, so what it represents and how it works, and then the partnership with Byramjee. How do we market the test? How do we accelerate the accessibility of the solution worldwide? So first of all, the WHO guidelines. This data, I guess, is very known to everybody who is working in TB, but it's good to kind of remind ourselves where we stand today and where we are. You know, where we are. TB, as we know, is preventable, curable, but still it is the highest infectious disease killer worldwide. Unfortunately, again in the first place after COVID-19. Yearly, we have almost 11 million cases detected, 1.3 million deaths, 3 million go undetected. We are at the level of 400,000 NDR-TB, which are the drug-resistant TB globally, where 40% of drug-resistant TB go undetected. So we have new treatments, new repurposed treatments, new repurposed drugs. We see that in many cases, many regions, the resistance to these new drugs is rising. So that is a very alarming situation, meaning that, you know, bedaquiline, for example, took 10 years to develop and implement into the PCAL regimen, and that is one of the, you know, one of the key drugs today for TB treatment, and if we lose this drug, we lose again years in fighting TB. 30 of the top burden MDR countries harbor 90% of MDR-TB. So it means that the MDR-TB is very, very kind of essenced in only 30 countries worldwide. And unfortunately, TB thrives in limited resource settings. So how it all started? In 2019, organizations like FIND initiated the Seek and Treat trial, where we are also involved and invited. We developed the Ampore TB test, and we were included in this trial. Where no, I will not go into details how many patients were tested and so on. So the good news is that the trial was successful. We were compared with alongside with two other technologies. So one was the Illumina, or the duplex technology that you're probably aware of, and the second one was a manufacturer from China. And just recently, the publication came out 3 weeks ago. It was published with very, very nice data. I will just cover a couple of those points in the publication. And based on this Seek and Treat trial, the data was transitioned to WHO, and WHO was able to evaluate it and actually include it in the third version of the WHO recommendations. So it means that Ampore TB is included in the WHO recommendations, which is a huge milestone also for Oxford Nanopore Technologies, and a huge achievement. So, for those who have seen the WHO recommendations, might have noticed that not all the drugs that other technologies are included are also included with Oxford Nanopore. So, you know, namely, you can see rifampicin as nice with fluoroquinolones and other drugs, but you don't see bedaquiline, you don't see clofazimine, and similarly. So that is simply related to the fact that when we developed the test, we relied our knowledge on the database of WHO. So the first version of the WHO resistance catalog, and drugs like bedaquiline, clofazimine, and similar were not included in the or resistances were not included in the catalog. So what we were able to do is, when the second version of the WHO resistance catalog came out, we were able to include this into our software or analysis pipeline. We were able to reanalyze the already run tests by FIND, and FIND also reanalyzed it. And we were able to improve the resistance for bedaquiline, for example, from 6 to 70%, for clofazimine from 0 to 70%. So, which actually shows that this environment is very, very dynamic. So, sequencing sometimes can, you know, in sequencing technology, you can improve the performance just by changing a little bit the bioinformatics. We were not forced to go back and really change the wet lab of the test, because we already included the targets into the test initially. So that was a huge achievement, and from the timelines perspective, we were told that WHO is holding a technical advisory group meeting in January, and hopefully after that they will be able to, you know, release either a quick rapid communication or an amendment to the existing WHO guidelines, including those targets, which actually places up really head-to-head alongside with the existing technology on the market, and we can be very, very competitive. So just quick overview of the guidelines, as you probably know. So there's a whole bunch of recommendations. They are grouped in 3 main groups. you have initial tests for diagnosis TB with drug resistance detection, then you have the initial tests for the diagnosis without drug resistance detection, and we're kind of falling into the third group because we are the follow-up test, so the follow-up diagnostic test for detection of additional drug resistance. So initially you have the gene expert with RIF resistance, you have the mobile with RIF5H to detect resistance detection, and similar. So we kind of fall into the third group where we detect the additional drug resistance. So this is all in the WHO recommendations. And then how it looks like, so you have the technology class, they have stepped away from naming brands in their recommendations for obvious reasons, because the gene expert was the superstar for many years, but now other technologies are coming in. So they have the low complexity automated NAT, which the expert XDR cartridge falls in, and then you still have the line probe assays, and then the third group is the new group, which is the targeted next generation sequencing group, where the duplex is included, Ampore TB test, and the TB-seq test from Schenking Medical Technology Corporation. So that's a high-level overview of the WHO catalog and WHO recommendations and where we stand today. So now let's switch gears to the Ampore TB test, and like I said, so I will give you just a high-level overview without going deep into technical, but kind of give you a grasp what the test represents. So we are talking about 6-hour or the same day sampled result with 2 hours sequencing with 16 anti-TB drugs screened with one integrated solution. So we have also the test here. You'll be able to see how it looks. And just to give you an overview of how the workflows, the workflow looks is that, again, this is a reflex test for TB positive samples, so that's very important to kind of highlight. This is not a diagnostic test. We don't have the diagnostics claim in the test. So the sample is decontaminated sputum. We have validated automated and manual extractions in the workflow. And the kit comes with a reusable flow cell, which you will also be able to get your hands on later on. So the workflow is, so the test works from DNA, right? So you will still need to do the extraction part, the contamination part, the PCR in your lab, and then prepare the library. And the sequencing will take only 2 hours, which is a huge advantage opposed to other technologies out there, or the technology out there. And another advantage is also that you don't need any bioinformatics on the site. The analysis is done within the software, and you have a user-friendly interface and user-friendly result reporting, which I'll come to in further slides. So, like I said, the extraction we have validated. I think my colleagues will speak more about the extraction. So, automated extraction on the Promega Maxwell and the manual extraction on the QIAGEN kit. I will not go into details on that. So, this is the antimicrobial resistance that we are detecting. So that we have the 4 first-line drugs and 12 second-line drugs. So pretty much all the drugs that we have today, in the treatment, we detect. Additional targets what we have in the test is the HSP65 target, which is the NTM related target. We do not differentiate the NTMs. We just say that it is present in the sample. So if you see it present and you're interested more in NTMs, you can do that in the follow-up testing on the specific patient or sample. So I kind of prepared the next slide for one reason, is that as exciting as the technology is and as deployable as the technology is, it still has a lot of limitations, and we have to be very aware of that. Because if you look on the bottom side is the kind of the workflow that I was talking about, from decontamination through the extraction, PCR, and the sequencing, Ampore TB covers only part of that workflow, right? So you need to understand is that if you want to do sequencing in your lab, you still need a kind of significant infrastructure, expertise in place. You know, a whole bunch of instruments, cold storage, a whole bunch of additional items that are not included in the test and that need to be procured from a third party, and you need to train the personnel and so on. So we are still limited to central or maybe peripheral regional labs, but we are still pretty far away from point of care application. So this just kind of keep our feet on the ground and be very realistic and aware of where we stand today. Just a quick overview of result interpretation without going into details, because this will be covered later. So on the left side, the advantages of this technology is that it is a built-in workflow on the GridION instrument currently. There is no cloud connection or no internet connection required. All the analysis pipeline is on the instrument itself. It is automated. After the sequencing, you see the automated results. And we also have a snip and indoor callings, which Rebecca will cover in this. And so yeah, I mean, this is the interface and the results. Just to zoom in on the results on the bottom, you see. And in a nutshell, so we have a sample to result. It's a one-day sample to result solution with 16 drugs screened. It is a low or almost not a maintenance instrument for many various reasons, because effectively the sequencing happens in the flow cell that you will see later on, which is a consumable. So we want to recycle it, but it is a consumable, and the instrument itself is a big computer, if you want, powerful computer, which needs very, very low maintenance. It is very easy to implement. It can be deployed in the diverse environments, and it has a very small footprint. So for those who know how the GeneXpert 4-module instrument looks like, and I guess most of us in the room knows, it's pretty much the same size. So it's a benchtop instrument, a little bit heavier than the GeneXpert. Right. So and then just switching to the last here is I would like to touch upon our partnership with Byramjee. So a global partnership with the company Byramjee, and thank you again for being in the room and for organizing this for being part of this event and having us here. So as you might have heard, last year in April, we stepped into a partnership with the very well-established global IVD company called Byramjee. And just recently, a couple of months ago, we signed a distribution agreement for Ampore TB. So we have a couple of projects going on with Byramjee, but one of them is the distribution of Ampore TB worldwide. So why did we step into partnership with Byramjee for Ampore TB? For a simple reason, because Byramjee, the company, has a very strong international presence. They are located with subsidiaries in about 45 countries. They serve more than 160 countries through distributors and other networks. So it is a well-established network where we can pretty much use it as a vehicle to go to market. And this is the intention. What we want to do is to really accelerate the disbursement of the technology worldwide. So we're focusing on 3 channels with Byramjee. So the first 2 are more through subsidiaries and the distributors. So this is something, like private labs and commercial sales and so on. But what we are also doing is we are very focused on the global health part. So Byramjee has, let's say, my counterpart for global health, where we together are working really with organizations like the Global Fund, the WHO, GDF, all the pool procurement mechanism kind of platforms to be available for the public sector. Because as you know, TB in many countries is an issue, or in most of the countries actually, is issue is addressed by the public sector, and getting access to the public sector is very important to this technology, especially since we are recommended by the WHO. So, so this is something that we're working on very intensely. So it means that there are processes that need to be done, that need to be, we need to undergo and really have accessibility to the technology through those platforms. So with this, I'd like to finish my slides. If you have any questions, I'm happy to take any questions. Otherwise, I would like to give the floor to my colleague, Lamok Zaveri, who is the field application scientist for Oxford Nanopore Technologies, and will take over and give you a further deep dive on Ampore TB test.
Any questions in the house? Anybody would like to?
Regarding that NTM, can it speciate the NTM or just tell it whether it's present or not? The non-tubercular microbiome.
NTS.
Can it speciate?
No, so it doesn't differentiate the NTMs. It just gives you the presence of the target. The HSP65. It will indicate you that the NTM is present in the sample. Otherwise, it will not differentiate.
In India, there is a diagnostic, for a diagnostic, there is a guideline, National TB Elimination Program. What is the status of this Ampore in National TB Elimination Program?
Yes, so I think this is something that we will be addressing also together with Byramjee. I think we are very early, in a very early stage, I think, for this. But we are definitely, I think maybe Tushar, you could take this.
And we have presented, introduced this technology there. And what I see that there is an interest, especially for the pyrazinamide. They are looking for the solution. So now we were also in the evolution, so evolving stage. So now we are more formal is there in the guidelines. So we will be again talking to them, and we will further, try to reinforce our message of, because we are also doing, and the other technologies are also trying to get into the NTP program. So let's see in the future.
What about the extrapulmonary samples, sir? It's just for
Yes. So, yeah. So currently we have, the only sample that we have validated is sputum, because we are then afterwards going the IVD route, so we need to do the clinical trials, and obviously you cannot expand it. And it will be a similar situation that we had with the gene expert. Just to give you a background, I was 10 years working for the company Zipit, so I'm pretty aware how the gene expert kind of implemented. So I expected it will be a similar situation where many research groups will do extrapulmonary samples, and we will see publications coming out. It is just we are very early today, right? And we actually have one group, which is Baylor College, who does stool testing on stool samples, and very successfully. So we do hope that we expect that next year we will see some publication. And they actually had some preliminary data already presented through last week in the TB Union session. So it's exciting to see. But we will validate only sputum. Commercializing it together with Byramjee. We were looking at the target product profile by WHO. WHO kind of indicated the $50 threshold per sample, which we want to achieve, but it's not achievable yet, to be honest. About commercialization specifically for India, I think again Tushar might take over. But so I think we are better sitting as our competitor. I mean, it's still we cannot talk about $5 test per result like similar that it's requested by from Sophia. we still have limitations. And like I mentioned, you also have limitations in the infrastructure because you need all the infrastructure around it still, right? Maybe you want to add something?
So we are currently we are in negotiation with the Global Health and for the LMIC countries, and we will follow the Global Health pricing guidelines in India as well. So that's
So the global health pricing is more or less defined. So we are looking into $75 to $80 price by test by sample, but this is only if you use correctly the kit. It means that you use all the reactions, so this is somewhere there in this range we are looking into.
So in other words, we can see that we need an expertise on this. So that to run this, so we need an expertise who are going to provide us with the information and stuff. So you need a dedicated trained personnel to run all this. So that means an extra demand for us for the LMICs.
Absolutely. And this is why I said we are still limited to central or regional laboratories because when going more into third level, it's going to be difficult. But we're working very closely with Byramjee to really get them up to speed, and then the training initially in India will be provided by Byramjee as well.
So we will be building the infrastructure for training and providing all the I would say initial training and adopting this technology within your ecosystem. So and we are partnering with the technical partnership, commercial partnership with the Nanopore. So we will be providing all the support here through our team.
Last question, how much does a gridiron cost?
I don't have the price today, and I mean essentially it will be Byramjee who will communicating the price. The standard price that you can see online on the ONT platform is, I think, 75,000-ish euros, but we do expect that we will have a better solution for resource-limited settings through the global health as well.
I have a question. As you said that you were working with Cepheid, and now that Cepheid has launched the new Ultra XDR cartridge, and we are right now putting samples for XDR, and we have around more than 10 drugs which we can detect like the XDR. How different will this technology be in a government setting where we already have Ultra XDR for pulmonary samples, and we are also trying to do extrapulmonary, and also to get this technology, how beneficial it will be in a diagnostic perspective?
So first of all, XDR is limited to 8 drugs, if I remember correctly. So we have 16, and we have all the new drugs, including bedaquiline, clofazimine, and similar, which XDR doesn't have. Still, XDR can play a role because it can be deployed near the point of care, where our technology cannot be deployed near the point of care. But if you really want to see the whole kind of spectrum of antimicrobial resistance, then you would go for next generation sequencing. Today, there is, to my knowledge, there's no molecular test that provides you the whole area. Any other questions? Okay, so with this, thank you very much. Thank you for the interesting questions. We'll be here all day, and I'm looking forward to my colleagues taking over the floor. Thank you.
Yeah, much better. Okay. My name is Lama. I am the field application scientist for Oxford Nanopore, and I'm based out of India. And I'm going to give a brief overview of what Nanopore is all about and a brief overview of the technology. And to first start off with, the first question obviously is everyone keeps bandying about the word NGS. So the question is, what exactly is NGS? Okay. Now, when you think about NGS, okay, has anyone over here done Sanger sequencing? How many have done Sanger sequencing? Okay, we've got a few here, a few more. Okay, exome panel sequencing. Have you heard of exome panel sequencing? Yeah, okay, a couple. So the idea being is you're using genetic information to build a picture of what's happening inside a patient or what's happening inside a cell. So the idea of NGS essentially is taking that genetic information, putting it together to get an idea or a picture of what's happening in a body, a patient, a cell, an organism, whatever it is. So it's essentially like building a jigsaw puzzle. Okay? And when you think about jigsaw puzzles, I can give you two options over here. Okay? Now we've got super complicated small pieces look the same, and then on the other hand you've got those large pieces over there. Now I'm going to ask a very simple question. Which jigsaw puzzle do you think is easier to solve? Obviously the one with the larger pieces, and that's exactly what Nanopore Technology is about. Long-read sequencing. Essentially move away from the short pieces. I mean, that's fun. I mean, if people are interested in some challenging, let's do something interesting on the weekend kind of a situation, then fine with the short, small pieces and things like that. But let's face it, we don't have the time to do all of that. In our field, we want quick answers, quick results, and that's exactly what long-read technology is all about. And the question is, obviously, how does long-read sequencing work? At the heart of nanopore sequencing is the nanopore. Okay? This is a modified protein, essentially, which allows DNA to pass through this. And if you remember Ohm's law from back in college, V is equal to IR. So now if I were to put, now, if I were to put a potential difference across this, what that means is it starts to create a current. Okay? Now consider if I pass the DNA strand, there's a disruption in the current. Essentially, it's a resistance. By putting a fixed potential difference across, and by disrupting the current and by measuring the change in current, what we can do is we can correspond this particular change in current to the corresponding nucleotides that are going through this particular pore. And that's exactly what nanopore sequencing is all about, essentially. Where you take your DNA strands, you pass it through a pore and measure the changes in electrical current. And then this change in electrical current is by using our particular software can convert this change into electrical current to ATGC. And thereby what that means is this gives you a lot of freedom in terms of what you can do with this particular technology. The most powerful feature of nanopore sequencing is the fact that you can directly sequence now either DNA or RNA. In fact, nanopore is the only technology on the planet that can directly sequence RNA, which no other platform can claim to be. On top of that, a lot of sequencing which is based on PCR-based, which introduces PCR-based amplification, PCR-based errors, which is not something that we really want, especially considering that we're looking at SNPs and variants and things like that. So this is another challenge that's there in the field, and if you were to use nanopore sequencing, you would not have to worry about these kind of technical limitations because of the fact that you are directly sequencing the genetic material. Not only that, because you're using a nanopore-based technology, you're not limited by read lengths. Now, if you're done Sanger sequencing, you know you can get maximum read lengths, sequences of 500, 600 bases. Short read sequencing, 100, 200 bases max, but with nanopore, there is no limitation. It's just based on how good your extraction protocol is, you can go up to megabytes. In fact, one of the longest reads we've actually sequenced is 5 megabases, so that's an entire. That's a very, very powerful technology. And going back to the jigsaw puzzle, given a choice, would you rather be solving the Mona Lisa with the small pieces, or would you rather have the dinosaur puzzle over there with the large pieces, which will give you a quicker answer faster? Not only that, the other advantage of the nanopore technology is the fact that you can do real-time data analysis. And what that means is, while the run is going on, you can do data analysis. A lot of systems have a model where you put your sample in, you come back 6 days later, and you get your result. And that is something that you just don't want. You want to be able to analyze the data as and when, especially when you're considering diseases and diagnostics, because you want results now. If you can have the option of getting your results in 2 hours while the run is going on, so you can do quick diagnosis, would you like to take that? Or would you take the traditional methods like that? So that's how small this guy is. The means is we have a lot of flexibility in where you can use this particular technology. Others who've taken it from the sea. You've got this thing, a lot of projects that are also there in Antarctica, where rather than collecting samples in Antarctica and processing them somewhere else, you can actually do direct sequencing at site. And in fact, the nanopore technology has also been deployed in a lot of our outbreak areas like Africa and things where we set up camps at outbreak regions along with a lot of health organizations to do diagnostics on site. In fact, even in India, NIB uses the MinION at site. So when there was the Zika outbreak in Kerala last year, the MinION was deployed over there for all of these kind of outbreaks. So that's the advantage of what we use Nanopore. And that's just a brief overview of Nanopore. I'm going to pass the mic on now to Rebecca, who is going to actually be giving us a better overview of the entire technology and also the workflows and the software. Thank you.
Just a quick comment. Just coming back to my slide about the limitations of the technology. It is super exciting and it is great. Just when we're focusing more on the Ampore TB side, I mean, we need to kind of be focused also that it is a clinically applicable test. You will not be able to do it on a space station, right? On a minute, just for your information. Not that you will need to, but it's just you need to understand that it is a gridiron linked test for now. It will not run on the midnight. But thank you for the presentation. I mean, it is an exciting technology, and I think there's a huge potential going forward as well. Thank you.
I'll be focusing more on the Ampore TB and those applications with it. Again, my name is Rebecca Smith. I'm a clinical and applied field application scientist with Oxford Nanopore. I'm actually based in the United States, in case you can figure out from the accent. So thank you all so much for allowing me to come here and speak with you today. And hopefully my southern drawl doesn't come out too much as I talk. So I'm just going to go through a very brief overview about why. Why are we doing this test in the first place? So as we're aware, drug resistance TB is a rapidly growing problem. We all know this, that's why we're all here today. I'm very passionate in bringing this technology to new applications where we can help solve problems. My background is more clinical, so I'm very excited that we have this test that we can present to you all today. Really what we're concerned about is we've made really big strides with TB, and we don't want these drug resistance versions of TB to really reverse the progress that we've made so far with this. So what we've seen is, especially since 2019, the number of cases really have increased, particularly in certain regions of the world, and it has been the 13th leading cause of death worldwide. So we really want to figure out what can we do to combat this and really make sure that patients are getting the correct treatment fast. So we're all aware of what the current gold standard is and the problems with that. So currently, the majority of the testing done is with culture. We all know that this will be very slow to get your results. It can take anywhere from about 4 to 6 weeks. This is very centralized. It's very accessible for a majority of the samples. Flip side of it is, we know molecular testing can also be very limited to just a handful of markers. You've got CapEx, it's very expensive, and that could be cost prohibitive for a lot of laboratories to bring this on. So because of these two issues, it takes a long time to get the correct treatments. It takes a long time to get started on the correct treatment, and unfortunately, not that many patients are able to survive long enough to get the correct treatment. So at Oxford, what we have done is try to figure out a solution for this problem. So with Ampore TB, and again, this is going to be eventually an IVD product, so that's why we have certain limitations with it right now, but we'll just kind of talk through the test. So with our solution, we have it being derived from sputum, so that way you don't have to mess around with culture and wait till you get those results. I'm not going to go too much in cost because I'm tech support. I'm not sales, so I'll let the rest of the majority of folks answer those questions. But really with this particular test, what we want to do is have a same-day turnaround, which would be fabulous when you're working with drug resistance tests, and then also you're working with things like sepsis and things of that nature, because we really want to be able to give the patient the correct treatment fast. So with this particular test, you're able to get this done within the same day. It is a comprehensive detection of all known and validated drug resistance markers. So again, we do follow the WHO guidelines. It can match the sensitivity of culture and other PCR alternatives. And ideally, we would love to be able to get this more into decentralized and other local laboratories and clinics. So we break down a little bit more with what's in the kits, and I apologize, it might be hard to see all the various different targets that we have listed here. But currently the kit is research use only, so I have to kind of give you this disclaimer with it, but we are moving forward to get IVD regulatory approval for this. How this kit works is we amplify and work with 24 different amplicons, so that's going to cover 24 different targets. That's going to provide all of our drug resistance information. So within those 24 targets, we look at over 1,200 drug resistance associated mutations. All this can be detected within a single assay. And what's beautiful with this assay is if there's any changes to the WHO, we don't have to make any changes to the wet lab portion of it. All we have to do is just make changes to the bioinformatics side of it. We are looking at first and second line anti-TB drug resistance in there. So basically we're going to identify those MDRs and XDRs with TB. And also within the kit and the additional targets that we look at, as we mentioned earlier, we have the HSP65 target in there. We have our spligotyping target, and then we also include an internal control. So we're going to break it down a little bit. Again, we've already kind of seen a similar slide earlier. But right now, this has been validated on decontaminated sputum. So the sensitivity is roughly at about 350 genome equivalents, or essentially about a CT value of 33 or below. Anything above 33, you might get some data or just might fail completely with that. Again, I've already previously mentioned, we have about 24 amplicons, which are going to be targeting 24 different genes that are going to cover those 1,200 drug resistance associated mutations. And sequencing only takes about 2 hours on the GridION device. So again, turnaround time is going to be within a day. What's really nice is that you can reuse the flow cells. You can get up to about 4 uses with that flow cell. So essentially you're going to be running a total of 96 samples with controls. So that's going to help cut down on your cost. And what's beautiful, once you're done with your sequencing portion, it automatically moves over to the data analysis portion of it. So there's no need to hire an additional bioinformatician. It's just going to move straight to that analysis protocol. So this takes roughly, and again, marketing put this together. So we all know, realistically, for those of us who have worked in a lab for a while, these times might vary a bit. But typically it takes roughly about an hour for you to do your decontamination, cell lysis, move forward into your DNA extraction, purification. Then you have your multiplex PCR portion, your quick library prep, and then our sequencing. So there is some hands-on time during this, but there's also a lot of like walk away work and other things as well. I'm not going to go through every single one of these, but you do need to provide additional equipment and consumables. So anybody who has a molecular lab typically already has all of this equipment on hand, so this shouldn't be too surprising. Again, just a typical magnetic rack. We do recommend a qubit with it, the devices that comes with it, and then just additional, I do want to point out, you want to make sure you're using low-lying tubes with this test. Okay, so reagent-wise, we really tried to make this a little bit simpler for you so you're not having to buy a ton of reagents, because I know that can be challenging to get certain reagents in. For the sequencing side, you just need to have the sequencing kit, the flow cells, and your recalibration kit, which is going to be used to allow you to reuse your flow cells. And the only miscellaneous reagents that you need to purchase or have on hand is just ethanol, and of course the reagents for your qubit. Okay, so before we start talking a little bit more about actually how the steps of the process of setting up this test, I do want to point out a few notes for you that's with the kit. So we do include our assay controls within this kit. So you're going to have your positive control, You're going to have your no template control, which is also, some folks might call that your negative control. It's interchangeable. And then you also have your internal control as well. So for every 22 samples, you do need to include one positive control and one negative control within that. So the positive control, that's going to be, is provided with the kit, and that is going to be included at your PCR stage. And so this is going to be treated exactly like your samples throughout the whole protocol. So this is also going to go through sequencing as well. Your no template control or negative control, we're going to include this at the extraction stage. So this could be either nuclease-free water or PBS blank, and it's going to be treated exactly like a sample throughout the process. So none of this should be too surprising if you do any type of molecular testing or diagnostic testing. And then lastly, we have our internal control. So this is added to every single sample along with the control tubes. It is, I want to make a note here, a competitive control. So it should only amplify in the absence of TB DNA or if the TB is below the LOD. It is a synthetic piece of DNA and it will not align to the TB sequence. And it is very, very, very important that you do include your positive and no template control in the test. Otherwise, when it goes to the data analysis portion, the results will be invalid. Okay. As we previously mentioned, this has been validated with sputum samples, but we've had other users in the field who have tested other sample types and have been successful with that. The samples must be decontaminated prior to extraction, otherwise that will affect the quality of your results. If you have any frozen pellets that you're working with, you just need to resuspend that pellet in PBS. And again, only up to 22 samples were processed at a time. For DNA extraction, these are just the two methods that we have validated. By understanding and working with other labs around the world, there are labs use completely or different methods, and that's fine. We'll just need to test those out with the kits, but these are just that we have tested internally. So we have manual or automated solutions with the license portion of it, depending if you're doing manual or automated. You have to do either the fast prep bead beater or vortex. For our manual extraction, we have validated the QIAGEN DNA Easy blood and tissue kit. And then for the automated portion, we have validated the Promega using their RSC Pure Food and Pathogen kit. With master mix, so when you're processing, and this again, this shouldn't come as any surprise to you, but when we're working with multiple samples, it's always good molecular practice to include an excess of about 10%, just because you're going to have that pipetting error in there. And this has already been calculated into volumes. This is already more than enough reagents in the kit, so you shouldn't run out of any material as well. And then again, for contamination purposes, to cut down any potential contamination within your laboratory or as you're setting up this test, it's always good molecular practice to have a pre and a post PCR area. So we're going to be doing our extraction and PCR setup in the pre-PCR environment. And then once we've completed that portion, we're going to move everything over to our post-PCR area to complete that library prep and sequencing. Again, just basic recommendations, make sure you're wearing your PPE at all times, make sure your lab bench is down, your pipettes down, etc. when you're done. So again, we've already mentioned this a few times, but this is just kind of like the recommended protocol that we have put together. We're going to go through, do our decontamination portion of it. One thing that's very important to note with the decontamination piece is we can process up to 700 microliters of sputum samples. If you go with a higher volume, it could overflow the extraction column, and that can lead to inefficiency of your DNA extraction. So it's very important that you're only working up to 700 microliters. Once we do our decontamination, we're going to heat kill and then lysis, and then from there you're going to move over to your extraction method. So again, the two different methods that we have validated will be either the automated extraction method using the Promega, or we have our manual extraction method using the QIAGEN kit. So this is just want to reiterate, if you go with the automated extraction, you do need to use the feed feeder. That is part of the protocol to use that. And then if you're doing the manual extraction, you would just vortex your sample instead for that portion of it. All right, how are we on time? Good. So what I'll do is I will move forward into talking more about the library prep. And what you're going to realize as I talk through this, it's a very, very simple library prep. I don't know if anybody's worked with Oxford Nanopore before or with Illumina, so the prep's going to be a little bit different. But the first thing we're going to do is our one-pot multiplex PCR. So super easy. If you've made a master mix before, this should be no surprise to you, but essentially you're just going to go through, make up your master mix. You're going to do this in a pre-PCR area. You want to make sure we haven't, you just don't want to avoid any type of contamination. So we'll be at reagent-wise, we're just doing our primer mix, our GC enhancer, and our platinum tack. So it's nothing fancy that we're putting into our mix. And so again, our volumes are already calculated out for 24 samples, plus that overage in there. Once we have made up that mix, then we're going to add in our internal control. And again, the internal control is going to be added to every single sample. We're going to vortex and spin everything down, and then we're going to aliquot all of this into a PCR plate. So everybody gets the same amount of master mix, and then just depending if you're adding in your extracted sample or the controls will be a slightly different volume for all of those. Once we're done, we just cover the plate, seal it, vortex, and spin down. From here, we just go through traditional PCR. There's nothing fancy here at all. The only little things I want to point out is we do want to make sure that the lid is heated, and then if you do have a thermocycler that has a ramp rate, please use that. If you don't, it's not the end of the world. You'll still get good results with it. And then this is just an example on the right-hand side of what a plate layout should look like. So you've got your samples, and then you've got your positive control, and then you've got your NTC. Okay, so now we get to the fun stuff. Now we get to actually go into the library prep portion. So we're going to be using, if anybody's worked with Oxford kits before, we utilize the rapid barcoding kit. So this is going to take roughly about an hour to finish up your library prep. The rapid kit is a transposon-based chemistry. So basically what we're going to do is it's going to go in and cleave your your amplicons at various different sizes. It's going to attach on our barcodes, and as it's going through that process, we then will pull everything together, do our spry cleanup, and then attach on our sequencing adapters, and then move forward into loading. So it's a very, very easy and simple process to do. So again, get my pointer. Essentially here we have our amplicons, and so then these are roughly about 1 kb, and I realize that it might be hard to see the various different colors that we have here, so I'll try to point it out. So we have our bar-coded transposon complex. So essentially what this guy here is going to do is it's going to randomly cut and cleave your amplicons, and at the same time it's going to attach on these barcodes. So once we've completed this process, and this is a very quick process, it only takes a few minutes to do this portion of it, then we pull everything together. We do a 1-to-1 spry cleanup, and then we add on our sequencing adapters here, so these will attach onto these ends, and then we're ready to move forward into sequencing. Very easy. So this is what the kit is going to look like, and we actually have a mock-up, or I should say the sample kit is over here as well if you want to take a look at that. So what's great with the rapid kit is all of your reagents are included with this. You don't have to buy any other additional third-party reagents. So you're going to have all the reagents that are required for, I'll talk about a little bit later, for priming your flow cell. You've got in your NTC and your positive control, all of that's been included, and then all the various different other reagents that are needed to complete this process. Each kit is going to include all these reagents, so it's going to have everything for your PCR application, prep, flow cell priming, and sequencing. And again, each kit is made to work with up to 96 samples, so that's going to be your samples with your controls at the same time. So roughly that's about 88 samples and then 8 controls with that. And there's 96 different barcodes in there, so we don't have to worry about any type of crosstalk with those. Okay, so once we have done our PCR, so essentially what we're going to do is move it forward into our library preparation. So this is just kind of breaking down, giving you a nice picture overview of how we do this. So we have our PCR plate. So this is everything that we did with the first step. And then we have our barcode plate here that has a pierceable foil seal. So what we're going to do is we'll take 77.5 microliters of our PCR product, transfer that over to a new PCR plate, and then we're going to take 2.5 microliters from this barcode plate, add that together on this new PCR plate, and then we just do this very quick incubation step here, where essentially we just incubate for 2 minutes at 30 degrees, and then 2 minutes at 80 to essentially deactivate the enzyme. And then we cool that down to 10 degrees. Once this is completed, we're going to take everything out of this barcode, or this PCR plate, pull it all together into one tube. This will be our low binding tube here, so again, it's very important we're using low binding. We're going to go move forward into our scribe cleanup. So we're going to complete that portion. That portion takes roughly about 10 minutes to do. So what we do is we add in our scribe beads to this PCR tube, mix it up, and then we'll let that sit, and then move it forward onto the magnetic rack here, because we need to pull those beads to the side, and then we'll remove the supernatant. We're going to wash these beads with fresh 80% ethanol. And once this has been completed, we're able to then elute this out. After we elute it, we're going to pull it, or we're going to cubit this, because we want to make sure that we have over 50 nanograms of sample in there. And then we're going to do our adapter attachment in here. Okay, and I'll talk a little bit more about QC and the qubit in a second. So this is just, again, just a very quick overflow with this. So essentially what we do is we complete our final library prep. So we do add in a sequencing buffer and loading beads with that. So all those reagents, again, are with the kit. We need to then prime our flow cell, and I'll talk about that a little later. Why we prime, what's the purpose of that. So essentially we make our priming mix, and then we're going to add that onto the flow cell. Then we load our flow cell, and then we're going to run everything on the GridOn device. As we're sequencing, it's going to do our high accuracy base calling. So that will be happening at the same time. And again, this runs for about 2 hours to do 24 samples. And what's nice about the grid, we didn't really talk too much about that, is that you can run 5 independent experiments at the same time. So you can have 5 different flow cells running at the same time. All right. So the qubit. We do utilize the qubit throughout this process because we want to monitor and make sure that our test is working throughout. We do utilize the broadband double-stranded DNA kit, which is only compatible with the qubit 4 device. So we are going to qubit the positive control, or if you have your own control on there, you want to make sure you qubit that, and then you also want to qubit the NTC. And then we recommend qubitting 2 additional samples as well. So this is a good indicator as to whether or not did PCR work, because who wants to go through this whole process in library prep, and the next thing you know, like, oh, PCR didn't work, and then you got to completely restart this process again. So with our positive control, we expect that to be 20 nanograms or higher. Then with the NTC, it should be 5 to 10 nanograms lower than the PC. I do get questions on this. They're like, well, shouldn't it be much, much lower with the NTC? You have to remember, this is prior to cleanup when we're doing this step, and so you're going to have those leftover primers and stuff that's left over for PCR with that. And then with the positive control, we spike in just enough positive control to be at the LOD, so that's why it's going to be much, much lower. And then, of course, if you incubate your samples, they should be much higher than your NTC, and they should be much higher too. Usually they're much higher than your positive control. If these are not within the expected ranges, then we recommend that you go ahead and quantify all of your samples. And again, at this point, you can determine whether or not you need to repeat that PCR. Okay, then we're going to move forward into, again, we kind of already pre-mentioned this, and I'll talk a little bit more of this when we actually get into the demonstration of working with the flow cells. But essentially what we need to do is we need to perform our flow cell check. Then we're going to make up our priming mix. I don't expect you to memorize these numbers. These are all going to be in the SOP. But essentially, there's going to be different components on the flow cell. And I'll point this out when we talk about the flow cells. But essentially, there's two different ports where you're going to be, one, making sure you want to remove any air bubbles. And I will be harping on this a lot today because with most technologies, bubbles are bad. So we want to make sure we remove that. Then we're going to add in our priming solution that incubates for about 5 minutes. And then we're going to, during that 5-minute time, that's typically when I recommend customers finish up your final library creation. So that's where we're going to be adding in our sequencing buffer and then our library beads with that too, because as it's sitting for 5 minutes, it doesn't take that long for you to complete the rest of this library prep. And then finally, we will open up our spot-on port. So this is the second port that we have on there. And then we'll load in a little bit more of that flush buffer because we want to prime that port. And then we'll immediately add on our library in a dropwise fashion. And I will explain that in a little bit later why this is extremely important. And then from there, you're able to move, close everything up and then move over to sequencing. So before we move into flow cells, I want to address any questions you might have about the initial library prep portion, and then I believe we have a coffee break scheduled, and then we'll come back and move into the flow cell portion. But first I want to answer any questions you might have about the wet lab part. Usually it's pretty straightforward with setting up the library prep. It's very easy to do. No questions?
Everybody is conversant with the terms that she used. I think, Rebecca, you can expand on a few terms like adapters, barcodes. I'm not sure if the audience knows what is the function of a barcode or what is the function of adapters, what is library.
Yeah, let me.
You know, so these are the few basic terms that we use for any NGS work. So I think we can just a quick definition for each term.
Yeah, let me pull up a good picture of what we were showing earlier. So, I'm sorry, I'm a visual person, so that's why I like pictures. Yeah. Okay, yeah, so kind of breaking it down a little bit, like why do we need to do a library prep? What is the whole purpose of doing that in the first place? Because if you don't, you know, because my background is Sanger sequencing, so it's a little bit different with the prep that you do with this versus this type of library prep. So essentially, and I'm going to use terms in regards to the Ampore TB, so we're going to be talking more about our amplicons. So as we go through our PCR, when we do our multi-pod PCR portion of it, we're sequencing or PCRing up 1 kb amplicons. Great. We have these 1 kb amplicons, but what do we do with it now? We can't just go ahead and move forward into sequencing, because what's going to happen is, one, it may not even sequence because there's nothing attached onto it, what we call an adapter or this motor enzyme portion of it, or it's just going to go right through this nanopore way too fast, and we're not going to be able to distinguish what exactly the data is with it. So this is why we have to move into what we call library prep. So during this library prep portion, what we're going to be doing is adding on this adapter piece right here. So that's what this little purple guy is. And on this adapter piece also is what we have called a motor enzyme. So what this motor enzyme does, it docks onto the nanopore during sequencing, And it acts like a molecular ratchet and also like a helicase. So what it's going to do is attaches onto this nanopore. It's going to unzip those two pieces of DNA, because again, it's an amplicon, so it's a double-stranded piece of DNA. And then it's going to be able to control the speed at which this strand of DNA goes through this pore. So we do it at 400 base pairs per second. And we have validated this to where this is fast enough, where we can still get good quality data, and we're able to distinguish what this signal is in here. As we mentioned previously, what we have here is these nanopores are embedded into a synthetic membrane that has an electrical current here. And as anything passes through this nanopore, what this does is disrupts this electrical current and gives its own unique signal. So we're able to distinguish the A's, the T's, the C's, and the G's from that, and we can also distinguish if there's any type of methylation data on there, but that's if you're working with native samples. For what we're doing with amplicons, because we're PCRing, we're not going to have that methylation data on there. So that's just a little bit more background information for you. So during prep, I'll go back. Bear with me for a second. Lots of slides. Okay, so let's go back for a minute, and how do we multiplex 22 samples together and be able to distinguish the different samples on there? So this is where the barcoding portion comes in. So with barcoding, we have 96 different barcodes. So each of these barcodes are a small piece of DNA that has its own unique sequence on there. So what we need to do is, in order to multiplex these samples, we need to attach this barcode onto each one of those amplicons. So that way we can tell who's patient A, who's patient B, who's this patient on there. So that's how this library prep portion works. So on our rapid chemistry, again, we're doing transposon here. It includes these individual different barcodes on this transposon complex. So in each one of these different wells, when we go through and do our 24 samples, 2 controls, 22 samples, we're going to have barcodes, we'll say 1 through 24, and each individual well, when we do this complete complex, we'll say like well 1 will have barcode 1 in it. So that's what this complex is going to include on here, is the reagents to cleave your strand of DNA, and it's going to have barcode 1. Then in well 2, it'll be the same thing that's going on, but it's going to have barcode 2 on there, et cetera, with that. So when we go through and cleave each one, it's going to attach these individual different unique pieces of DNA onto your amplicon on there, and that is how we're able to distinguish between each individual samples. So we have 96 different barcodes, and again, I'll talk about this a little bit more when we talk about recalibration on there. But we want to, because this is going to be a clinical test, we also want to make sure that we're safe or we're not mixing up various different samples on there. We are only doing up to 96 different unique barcodes per flow cell, because we want to make sure that we are not mixing up any patient samples with this as well. So this is why we've got the 24 different samples, or sorry, 24 different barcodes at a time. So that way we can make sure we're distinguishing between our various different patients and our controls on here, and we're not mixing up any patient results with that. Okay, is there any other questions around that, or would you like me to go a little bit deeper into anything?
Of this test?
So the limit of detection with this is still being determined, because it is an RUO test right now with it. So again, this goes back to that CT value from the beginning of the portion of it. So it's roughly around a CT of 33 and up is what we're able to detect. So if you're 33 and below with that, if you're 33 and up, there is a potential you might get a little bit of information with that. If not, or it just might fail completely with that. So it's roughly around, I think it was 385, I want to say, genome equivalent. Let me go back. But we'll have more information, or exact 350 with that. Genome equivalence is essentially roughly what we've seen so far with the LOD, but again, it's still an RUO test, so once it becomes IVD, then we'll have it all that, essentially locked in stone with it. And again, this is really meant to be a reflex test. This is not meant to detect it as well at the same time with it.
One thing I want to ask, you have mentioned that there is a competitive control one out there that should only amplify the absence of TB DNA and or TB below LOD.
Right. Yes, we do include the control in there. The control, as I mentioned earlier, is a competitive control, and so that's the purpose of it. So most of these samples, since they should be TB positive, and so they're going to come in and use up all those resources before that internal control can go in there and use up the PCR reagents that are there. So that's why all of those markers are going to amplify, whereas that internal control is not going to amplify. Now, if you've got the vice versa of that, where it's a very low LOD sample or there's just no TB there at all, that's where our internal control is able to come in and use those PCR reagents in there and actually amplify.
And means you have one internal control which is different and competitive control which is different.
No, I apologize. The internal control is a competitive control. It's the same thing. So if it's a positive sample, the internal control should not amplify. If it's low or below the LOD or no TB there at all, the internal control, which is the competitive control, will amplify. Any other questions?
Is there any limitations of qubit devices with one kit?
Pardon?
Qubit devices.
So the qubit device that we've been utilized is the qubit 4, but I know there's various different versions out there. We just haven't validated and tested all of them, so that would be something you would have to test in the lab.
Presently, this kit is IVD. It is not an approved IVD.
It's RUO, yeah, it's not
It
IVD.
is under only the research use only. So can be used for a diagnostic test because the kit should be an IVD.
So that's always the million-dollar question with that one. I'm not familiar with your regulatory requirements here in India. I can only speak to the United States.
It must be IVD approved if you are doing the diagnostics.
Yeah, that's what we have to do in the US too. You have to treat it like an LDT and do a full validation of it.
Variety for NGS testing is
But that's the plans for 2025. That I'm working actually on the US side because we're going to be pushing it through the FDA portion of it to get an IVD with that. So both the test and the sequencing device will both be IVD. Any other questions? Okay.
When you spoke about barcoding, in other words, we can say that barcoding allows the multiplexing of multiple sequences in one run.
Right, because otherwise we're not going to be able to distinguish between the various different patient samples, and we want to make sure that we're calling the correct diagnosis or the correct drug treatment for each patient. So yeah, that's the importance of doing the barcoding with that. So each barcode is a very small, unique piece of DNA that gets attached onto your samples. They are not, these unique pieces of DNA don't match TB, they're completely different. It's just a completely random sequence that we put together. Many other companies do something similar, like Illumina does something similar with their barcodes. Yeah, and essentially what it does is when we get to the data analysis piece, it's able to, or when it's sequencing, it does the base calling with it. It can be like barcode one, ABA, BB, or AAA, TTC, whatever it is, whatever the barcode is. they'll be like, okay, this is barcode 1, and it's able to then move all that data to one folder, and then it can distinguish like, oh, this sequence here, this is barcode 2, and it'll move everything to that folder, and so that way when we move to data analysis, it's able to then just pull this patient's data in, run it through the analysis, and the next folder is going to have just that patient's data, move it to the next folder with that. Yep, and I'm around if you have any other questions if you don't want to ask them.
No, the entire day is there for them to interact with you and ask their queries. For now, we'll halt for a tea break, and then we'll come back and continue with the...
Yeah, we get stuff on first. Yeah,
so to remind you, we have also attached a small crossword puzzle for you. That's a fun name for you. So find time to search the words and solve it. Anyone who solves it first or gets the maximum number of correct answers gets a prize at the end of the workshop. So, yeah. We'll wait for 5 minutes. Once the tea is ready, we'll move to the third floor. This is the fourth, yeah. So we'll move down, we'll go to the third floor, have tea, and then come back and continue with our rest of the session.
And then what is the purple portion, the purple piece that's sitting on top of it? The adapter. Correct. And what does that adapter do? Yeah, that's part of its job. What else does it do?
Unzips the magazine.
Yep, unzips it. And then what other thing it does? Yes, those ones. I feel like a treat. Okay. What is multiplexing?
Different drivers, creation. Targeting the different genes and targeting every gene at the same time.
That is part of it when we're saying a multiplex PCR. I'm talking about with your multiplex, when you're putting the answer, right? So in this regard, so yeah, we are doing multiplexing with PCR because we're looking at 24 different targets within there, but we're also, when I talk about multiplexing, we're able to run multiple samples together on one flow cell. So how are we able to do that? What's the one little tool we need to use to be able to run multiple samples on one flow cell? Barcode. Perfect. Right. Oxford Nanopore. So with Oxford Nanopore, we have different types of base calling. Actually, let me go back on the slide. So base calling is essentially when we are taking So when we go through and this piece of DNA here goes through this pore, crosses the membrane, it disrupts this electrical signal, and what we have is the squiggle data. Wonderful. What do we do with it? So what we have done is we've put together an algorithm that's able to then take this squiggle data and determine what this pattern is and make it, is it an A, is it a T, is it a C, or a G? So we have different speeds of base calling that we're able to utilize during sequencing. So with Oxford Nanopore, we have what we call fast base calling, which essentially what it sounds like, it's fast. So the beauty with that one is you're able to get quick base call data, but you are going to sacrifice your accuracy, which therefore your error rate is going to go up with that. I don't have the stats off the top of my head because we do use a different pore chemistry with this, but I believe it's in low 90s, I want to say, so it's about 90% accurate, roughly. Don't quote me on this because I'm trying to remember this data. We utilize, with this test, our high accuracy base, so that's going to be your mid-level with it, so it takes a little bit longer to do the base calling, so basically turning this squiggle data into the nucleotides that we're looking at. That one is going to give you a higher accuracy with it, so I believe it's around 96%, 97% with the high accuracy with the R9 flow cells. And then we have our super accuracy base call model, which is going to give you a higher accuracy level, so a lower error rate with it. But the trade-off is it takes much longer for us to be able to base call this data because it takes more computing resources to be able to do that. So we have validated with this test that the high accuracy base caller that's with it is able to give us the good quality data with that and the correct results without sacrificing the time to do that with it. So roughly, I'll have to look it up for what it is for the R9 flow cells, but it's about roughly, I want to say, in that 97% accuracy rate with that one. And I apologize, I was going a little bit of the nanopore history with base calling. All right. Any other questions? If not, what we're going to do is we're going to move into flow cells. So flow cells are basically what we utilize to put our library prep on there and then be able to move forward into sequencing with this. So I'm going to spend a lot of time talking about these. So we utilize what is called our min-ion flow cell. And that's just the flow cell that we're going to focus on today. We do have different types of flow cells, but we do not use them with the Ampore TB, so I'm not going to cover those today. So with our flow cells, this is obviously much, much larger, and in a little bit we're going to pass flow cells out so that way y'all get to see what they look like. We'll get to practice loading with those as well. So this is what the top portion of the flow cell looks like. On it, we have here, basically this little snake-looking guy here. This is your waste channel. So this is where any type of used reagent is going to be moved into this location. We have two different ports here, port 2 and port 1, where we're able to remove any waste. And I will talk about that a little bit later because it is very important when we want to reuse your flow cell that we will need to remove the liquid that is in there. We have here is our priming port cover. So essentially what this does is this is going to cover an access port where we're able to input our priming solution. We have here what we call our spot-on activator. So basically this is another area where this is just a cover that's going to be covering on another port on the flow cell. The flow cell ID is located here on top. This area here is essentially the sensor array. So this is where all of the pores live. And this is, right now this is a yellow color because this has our storage buffer on there, but this color here will change when we add on our library prep. And then this section here is when you take the flow cell and flip it over. So this is, you won't see all of this portion of it because we do have a black piece of thermal tape that covers this, but this is what's hiding underneath that piece of tape. So we have our ASIC chip right here. So essentially what this is, ASIC stands for your application-specific integrated circuit. So essentially this is going to contain all of the electrodes and essentially the channels that are needed to run the sequencer or control all the individual different pores in there. And then lastly, we have here your connector pins. So these pins are what's going to attach to the sequencing device and be able to heat up and cool your sequencer as you're going through. We do have a picture later on, but just so you know that this is this works as a kind of like a unidirectional workflow with this. So when you add in any reagents here, it's going to move through channels, through here. It's going to push this liquid out, any type of liquid that you have here, out, and then it's going to come through into your waste port here. So it's all kind of got to move through in it. Okay, so to break it down a little bit more, when I was talking about earlier how we do a flow cell check prior to adding in our library and moving forward into sequencing, the reason why we do this is we want to check how many active sequencing pores, those little nanopores, are actually on that flow cell before we move forward into sequencing. Because otherwise there's no point in adding your library if you've got no pores on there to sequence with, and you're just going to end up wasting your time with it. So essentially how this works is each flow cell is going to contain individual wells and electrodes. So this is what a single well looks like on the array. And above that, you have your two different electrodes. So you're going to have your negative and your positive charges here. And as we all know, DNA is negatively charged. So when we turn these guys on, the DNA is going to want to move through the core towards this positive charge here. And then this is the membrane portion right here. And then each one of these wells should contain just one single nanopore. I do want to put a little asterisk there that sometimes during manufacturing, it could occur where maybe 2 or 3 additional pores have been inserted into this well. Maybe no pore was inserted into this well. We don't know until we do a flow cell check. So that's the importance, another reason why we want to do this check, because we want to know what's the best well that we can utilize for sequencing. So to break it down a little bit further, 4 wells are going to belong to a single channel. So when we talk about having 512 channels, that is what this is referring to. So essentially within each one of these channels again, we have 4 wells, and what we're going to be doing during the sequencing, or when we go through and do our flow cell check, or what we do flow cell check during sequencing, which is called a muck scan, and I'll talk about that a little bit further. But we want to check which one of these wells has the best pore to move forward with sequencing. And so this is also kind of like built-in redundancy that we have in here. So when we do our flow cell check, what we do is we go through each individual well and check to see, is there a pore? And is it a pore that we can utilize for sequencing? If there is no pore in there, we actually do our sequencing, that well will be permanently turned off during sequencing. We'll never utilize that one. Or if there's a well that has multiple pores in there, that's also going to be turned off during sequencing, because you don't want to have multiple pores sequencing at the same time, because then we cannot distinguish what that sequencing data is. So we want to make sure we're not utilizing those. So what we're doing is prior to any type of sequencing run, we want to test each well in each channel just to see what we have there, and then we measure that current that's in there and record that. And so from there we're able to determine, is this an active well that we can utilize? Is this one's inactive, meaning like maybe it's already been used too many times, we need to turn it off? Is it unavailable because there's no pore there, or because there's too many pores there? So this is everything that we're going to look through when we do a pore scan. And you'll be able to actually see this information, and I have slides for this later on in the presentation, or later on this afternoon that we'll go through, that's going to give you that information, so that way you can see how many pores do you have, how many are having healthy pores, how many are unavailable, etc. with that. Is there any questions about this part first before I move on? Okay. Yeah.
So you were supposed to have a synthetic solid state membrane, has that happened? They said it was in the pipeline.
It's still in the pipeline now. It's still a synthetic membrane that we have there. Actually, I'll talk a little bit more on the next slide about that portion of it, because it does affect your flow cell warranty portion with that. Any questions? Okay, so these are living biological material that is in the synthetic membrane, so therefore we do want to make sure that we treat them right and that we handle and store them appropriately. So all your flow cells, they are shipped to you in a sealed individual pouch. Don't ever freeze your flow cell. If you freeze it, because it is a synthetic membrane and you have the pores inserted in there, if you freeze it, we all know what for self-freezes, it shrinks. So it's going to cause those pores to pop out of that membrane, and that's it. We're never bringing those pores back to life, unfortunately. So it's very important that we are storing those correctly. The recommendations are you can store them for up to 4 weeks at ambient temperature, unopened. Otherwise, the best solution really to help maintain those pores are to store them in the refrigerator, and then they'll be good for up to 12 weeks. And again, because this is going to eventually be an IVD test, this information will be on there that these are going to be initially have the warranty period printed on there as well. And the same thing too, after you recalibrate a flow cell, meaning that's where we remove all the previous barcodes off of that so that way we can reuse that flow cell. It's very, very important to also store that in the refrigerator so that way we can get as many uses as we can out of that flow cell. And when we do that initial flow cell check, we want to make sure that it has at least 600 pores in there before we start the process. So to kind of backtrack a little bit about that, because remember we're talking about how there's 512 channels, why that 600 doesn't equal 512, how does that work out? So if we go back up here, again as a reminder, calibrate and store it again, you'll still have plenty of pores to reuse on there. I'm going to backtrack for a second. Priming a flow cell. So why do we want to prime a flow cell initially? So with priming a flow cell, it already has that storage buffer on there. And that storage buffer already has QC DNA on there that we utilize to be able to distinguish does the pores or does the wells have pores in there or not. Make a nice, good environment for sequencing. And the big thing too we want to add in is our tether. So that is what these little looking guys here, they're kind of almost like little fishing bowls in here. So we add in our tether, this is going to then drop down to that membrane and attach onto that membrane. And their job is to help guide our strands of DNA down to the pore and help them kind of go to the right direction. So that is their whole job. So these are not originally on the flow cells to begin with, so you have to add that on there. So I'm just going to back up to here. So again, when we do our priming, we just want to make sure that we're creating an environment that's more welcoming to loading on a library. We'll go through this when we go through the demonstration of actually doing the loading of the flow cells, but the biggest thing is too, we don't want to add any air bubbles or introduce any air bubbles on here. Because again, if you add any air attaches that membrane, or comes closer in contact with that membrane, that's also going to damage that membrane, which is going to cause those pores to pop out of the membrane as well. So we just want to make sure that we're treating these guys with lots of love and TLC. And so essentially what we're doing is, apologize if I'm jumping around a lot, but again, I am a visual person. So again, we don't want to accidentally push any air bubbles in, and then we're going to add in our priming solution. So again, these are just the priming reagents that we're going to utilize in the kit. Again, it's going to contain the ATP, because that's going to be important, because that's going to help fuel that motor protein, so that motor protein can do its job of going through, attaching onto the pore, and unzipping the DNA and maintaining that speed. So these are the two reagents that are in the kit. You have your flush buffer and your tether. Very mix these two together and refreeze them. So this reagent does come as a larger bottle, so it's perfectly fine, because I understand it takes forever a day for things to thaw. If you want to initially thaw that first bottle and then aliquot it out, just do not add the flush tether to it, because it is not stable with freeze thawage. You want to keep the two separate. So it's very important that we do that. Right. So after we've gone through the first initial priming portion of it, this is where we're going to open up the spot-on port cover here, because here is where the actual library port is. So when we add this in, we want to help prime this port. So essentially when you add in your second aliquot of priming buffer in here, a little bit of the solution is going to be pushing out of this hole, and that's what's going to help prime the port, so that way we can go ahead and drop in our library. Because this is directly over the array, we have to do the drop by drop with the library. Otherwise, if you stick that pipette tip down into that spot-on port, you're going to end up damaging the array and damaging the pores and the wells that are in that location. So I'm going to show you this video. So what she's doing here is she is opening up that priming port, which now gives us access to this area here. I apologize, it's going a little slow. And it's very important you don't pull up too much, because if you do, guess what? You just introduced an air bubble and you just killed that part of the array. Next, we're going to add in the priming solution, and you want to very gently either pipette it in or dial it down, because again, you want to gently, change the solution for these pores. And it's very important to leave a little bit of liquid into the tip. Again, you don't want to accidentally introduce any air bubbles. This sits for 5 minutes, so that way the pores can reacclimate to a new environment. And then once during that 5 minutes, that's where you can go ahead and do your library. That portion of it. This is where we open up now that spot-on cover, and we're going to add in that additional amount of priming solution. Again, making sure that we leave a little bit at the edge of the tip. And I'll play this video again for you. And once that's done, because again now we have primed our spot-on port, we're able to then add on our library, which is what the user here is going to do, drop by drop. And it should just immediately be pulled into the flow cell. Once that's completed, we're going to close all of the covers, because again, we just don't want any evaporation occurring during the sequencing. So I'm going to play this one more time. No, there's no marker. So typically what you want to do is just pull back to see about 10 to 15 microns. Again, this is just an arbitrary number, so you just want to see that you have just a little bit at the bottom of the tip that you pull up. And again, the same thing when you are hyping your solutions, and you want to again make sure you're just leaving a little bit at the bottom of the tip. And all of these, we have a lot of great training videos that are also available on the Oxford Nanopore website. You don't need to have an account to be able to view these videos, and this video is on that website too, in case you ever want to watch it again. See in the waste channel how you've got liquid that's in there now, so it's very important to, and I'll talk about this a little bit later when we go through and do our recalibrations. It's very important that we remove all that liquid that's in that channel, so that way you can reuse that flow cell. Because what happens is when you store it back in the refrigerator, it will start to evaporate in there, and then that storage buffer or whatever reagents is in there is going to crystallize, and that's going to end up clogging those channels that's on the flow cell, and then you're going to have a really hard time putting on an additional library prep on there. So it's very important that we're going through and we're removing the buffer. So moving forward, I've just got a few more slides, and then we're going to get into the fun part, playing with flow cells. Because again, as I previously had mentioned, you can reuse these flow cells, and that's good because we want to reuse these flow cells. Sales doesn't want me to tell you to do that. I'm going to tell you to do it because we want to be able to get as much usage as we can out of these. So this is why we're going to move forward into flow cell recalibration. So this is where we're going to remove any of that previous library that's on there, because we're only sequencing for 2 hours. So you still have a ton of library that's still sitting on top of that membrane. So we want to, and we need to remove that in order to be able to use this flow cell for another run and get more data. Because otherwise, if you just put in your next library on top of the old library, and I have a good diagram that I'll show in a bit, you're going to end up sequencing that first library first, and you might not ever get to that second library in that 2 hours, and that would be a terrible thing to do. So that's why we want to go ahead and do this recalibration. So what we need to do is create up this mix and then remove that previous already run library in there. So what this recalibration mix is just a buffer with a DNA enzyme in it. That is all we're going to do. Is what we're going to do is chew up all of the remaining library that is on top of the membrane, because it's a DNA ACE, it's just going to chew it all up in there. It doesn't chew anything up that's underneath the membrane, and the reason why we don't have to worry about that is anything that's been previously sequenced no longer has that nodal protein on there, because that nodal protein actually falls off at the end of sequencing. So everything is just going to be still, that can be sequenced will still be on top of the membrane. So what we do is we need to create this buffer with our enzyme in it, and ACE in there, and then we are going to do this in 2 different aliquots. So somebody made a really good analogy with this once. It's like you're trying to have a really dirty table, and you just splash some water on there, and you wipe it off and call it a day. You probably didn't get all the dirt off of it, so that's why we're going to do this 2 times. So we're going to add 2 different aliquots of this recalibration solution on there. We add 1 aliquot, it's going to sit there for 5 minutes, start chewing everything up. Then we're going to add in a second aliquot and let that sit for 60 minutes, and it's going to chew up anything else that's remaining on there. And then from there, we can either reprime the flow cell to move forward to running another library on there, or if you don't have any other libraries for that day, you can add on our storage buffer, and that way you can go ahead and store that flow cell in the refrigerator, so that way you can reuse it for another time. But again, very, 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 very important is make sure you remove everything out of that waste channel before you store it, so that way you can have a good successful run the next time around. We talked about this, and we do it in 2 stages. this is, let me see if I can get you a good picture of this. Apologies, I don't have the good skills to like, yeah, 1% names. Expand this part. I know, it's back there. Yeah, it's that close. Yeah, that's fine. Okay. So this is a really good diagram, and hopefully y'all can see the different colors in here about why we want to recalibrate versus not recalibrating. So here, so over on the left-hand side is an example of if we don't do the recalibration. So we have our first library, which is in that dark blue color, over on the left-hand side, and then the person just immediately added on their second library onto the flow cell without recalibrating, and you can see we have a ton of that dark blue left in there, and you just have a little bit of the red. Well, that's a problem because that may never sequence in that 2 hours. And then you can see they kept doing it where we moved to the third run. You can still see we have that first library still hanging out there. Again, that's a problem because that's you're sequencing useless data at that point. So this is why we want to go through and do this recalibration step in here so we can reuse these flow cells. And you can see over on the graph on the right-hand side how the person ran the first library. It's in that dark blue color. We went through, recalibrated, added the second library, and now we are very successfully sequencing that second library in there. And then so forth and so forth. So that's why we want to make sure that we are doing this recalibration step with it. Okay. And this is just giving you, again, an example. So there will be a little bit of residual previously used library on there. It's about, 0.1% roughly, but that's at this point is going to be considered kind of like background noise compared to your fresh library on there. So you're going to have a lot more data from your fresh library than you really will from your previously run library on there. And again, this is why we use the barcodes, because this is also going to help us weed that bit of data out that we don't care about anymore. And so this is just showing you an example at the bottom, being able to do your various different runs with your recalibration and move forward with that portion of it. Just want to make sure I'm taking those. And then lastly, when we do our recalibration step, it is very important too that we are adding those reagents in gently as well, because during sequencing, because again, you're heating, you're cooling, you're doing stuff, you're going to create some air bubbles in there, and that's like when you're cooking, you start heating stuff up, you get bubbles at the top. Those bubbles at the end of your first sequencing run that you see on there, that's completely normal. We just want to prevent those bubbles coming together and forming one massive bubble, which then touches your membrane and damages the membrane and the pores on there. So we want to very gently add those reagents in there so that way those bubbles stay at the top of the plastic shield and don't come down to make a bigger bubble. Okay. Yeah, there's the two things you can take away from today is actually three. One, bubbles bad, so we always want to make sure we're removing bubbles. Two, always get that waste out of that waste channel. And three, nanopore sequencing is cool. So you can put those any order you want. All right, now we're going to get into the fun part now of actually playing with the flow cells. So what we're going to do is pass out flow cells and tips and pipettes around to everybody, and then we're going to walk around to the tables and we're going to work with you guys with playing with the flow cells. And I'm happy to answer any questions too as I'm walking around the room as well. So this one here. I guess I'll show them first because that makes more sense. Well, we got two of us this week.
for TB के लिए और TB is the first. हाँ.
Oh my God, it's too late. It should be us.
Very common. You need it? Anyways, it is here now.
So if you want, you can put your torch option. Can I add this? You can add this. Yeah, you can. This is the code. Yeah, keep it straight for perpendicular always. Yeah, always. No, don't plunge.
Let's say for 5 minutes, and then we move to the next phase is where and so these are used, and because it's just an acetate that's on there, it might come off, so don't freak out. So then we move to the next step is where we do 200 microliters into here. You should start to see a bubble up there, and then we move to so essentially what we're in practice.
So that's like a position, and then this flap. Okay, so this is the reason why I'm telling you about opening close is very important. When these are both closed, it's a sealed system. You open it, and now it's an open system. So this is very important, right? When it's sealed, you should always keep it closed so that it's sealed. It's a one-way channel, so it's going from that's the inlet. It's a very tiny hole there. The liquid's going from here to here. This is where the nanopores are. This is where the actual secrets are actually. I'm just explaining the overall thing and then I'll so the liquid's going from here. This is where all the sequencing happens. And once the sequencing is done, all the waste comes out from here, goes here, and you've got this little curve, snake like which is where the waste is collected. Okay. So now what is the problem? So this is let me shift these closer. When we set it will first look for that identity. If it finds that identity, it will identify that this is from yours, this is from yours.
Because now this is using so many sample means
Or informatics, this whole area is going to be, and then this should just be. Sometimes it's a little bit slower, but it's So there you go. So that's how you do it, and then you would go forward into sequencing like that. So I think all these have the space for those programs. But yeah, I'll let you guys play with this part. Back praise that when we do this will be on the device too. So
yeah, so the writing part is
this
on here. So we can do one.
We first have our initial
so just practice with the close up. Basically, get the idea of the concept of behind sequencing.
If you have these in here, you've got
NGS.
This port is like a door. So you have close, like your port is closed.
You're able to speak and you can the door. I want these boxes.
Let's use a lot. You can
let's use samples.
essentially you're able to go through. Yeah, because that's your only way to access this.
If you've done more bio or so it's essentially literally just one PCR, then 1 or 2 incubation steps, and then just put it on. All the steps that there are.
So the PCR which we
and these are you can see how we have these air bubbles here.
you mentioned it has to make sure it's starting concentration as mentioned before if it gets potential validated.
You can use any extraction set that your is preference. Yes. So the only one that we really ask is you do some sort of
That's brand new. Yeah, you first.
I'm going to do it inside the box. The thing is,
Yeah, so basically.
So this is the set scenario where okay, that's what also fact essentially. The idea being is to pause all the rest of 34 degrees Celsius.
You would go through.
So one of the jobs of this machine is this.
Do your flow cell check, and then you would prime.
So it's always kept at 34 degrees. And the pause also kept at 34. And also over here. Yeah. Okay. So the idea being is
That is a good question. So Linux is supposed to be a little bit more stable than Windows, and that's why we've designed the software off of that portion of it. And again, with this box, it's going to be more locked down, so you're not going to be able to install a bunch of different softwares on it.
Usually, as standalone systems for sequencing, you wouldn't want to mix it with anything else, otherwise also isn't it?
Well, yeah, usually, but with how our software and how we've designed it and validated it, and actually the grid devices are awesome. They are great little workhorses, so it doesn't take much computing power to be able to do the sequencing and the base calling at the same time. And then once the run's done, it moves forward into analysis. So even though you might have another sequencing run going, the analysis, the way the code has been written, does not take up as many computing resources either. So you're able to have all these different things going on at the same time and not worry about something crashing.
So the computing or power required, let's say, for a design versus a design, which would be more?
This one's more, but this is a bigger computer that's in here. Because we have different recommendations for a MinION, because we kind of have like base recommendations, like you kind of have like the bare minimum that you need versus the super duper computer that you can get for MinION. But again, it just kind of depends with that device what applications that you're looking for. Because you might be able to, if you're doing kind of like more like metagenomics work, that doesn't require much computing because those are not huge viruses and bacteria that you're looking at. Versus if you're doing human genetics, that's a different ballgame. It requires a bigger computer for those. Yeah. our run reports look like. So you're still going to get these run reports with it. It's going to kind of break down a little bit more information with it. A lot of this might not even be of use or interest for you. It's more so just to see what was the health of your sequencing run to make sure everything has been going appropriately. So I'll kind of break it down a little bit for each one. So underneath your run summary, it's going to give you an estimated number of bases that were sequenced, and it also gives you an estimated number of reads generated. So those numbers are going to be a little bit different because a read is that strand of DNA, and the bases are going to be based off of those reads. And the reads are going to be various different read lengths. We have the number or the amount of data that was produced, and then we have our estimated N50. I will explain what N50 is in a bit in case you've never done NGS. We'll get to that as towards the bottom of this run report. Then we can go down to where it says base calling. So that lets us know, did base calling complete? Hopefully it did. It should be at 100%, so that means every single strand that passed went through and was base called, and it tells you the number of strands that passed versus what failed. You will have some stuff that fails, it's expected, but it should be much, much smaller than what passed. If those numbers or those values are reversed, and you see a lot of stuff that failed versus what passed, that's usually an indication that something happened during your library prep or during that sequencing run. So unfortunately, you're probably going to have to repeat it. Over on run duration, it's just going to let you know how long did this run go for and at what time did it finish. Underneath run configuration, that's going to always tell us what type of flow cell you used, what was the flow cell ID on there, and what type of kit that you used. A lot of this information, all this is going to be fixed in stone, so you're not going to be able to make any changes with this. And this is really more so helpful for us on the tech support side, just to make sure that these values are correct on here. But essentially, it just kind of gives you a little bit more information. How long did it run for? When did the core scan happen? What was the minimum read length, etc. on here. Software versions just lets us know what software version you used on here, and again, just where everything was kind of stored. Just kind of covered this. Okay. So, read lengths. So, etc. in there is actually going to list the patient name or identifier that you gave it here, and then it's going to be actually listed in the order of your barcode. So, say for example, this Rep 1 for Zepto was your barcode 1, and then this is barcode 2, etc. in there. So that's going to list everything with it. And then it's going to list the number of reads that were collected with each one of the barcodes on there. And it's only going to show you the information for the barcodes that were used. And what you should see, which is also a good indicator of letting you know was this a good run or not, each one of these, where it says the reads, you should expect to get over 4,000 reads for each one of your samples, with the exception of your NTC. You're going to get some, because you always get some sort of background in there, but you should not see over 4,000 reads for your NTC. If you do, that's a good indicator that some sort of contaminant happened, or you mismatched and mislabeled your samples. This is the next little graph that's going to be on our run report, is our cumulative output. So this screen, or this graph here, tells you, this again, it's kind of probably hard to see with the lighting here, but with this darker blue line up here at the top, this is the estimated number of bases that we collected and base called on here. This green line here is everything that has passed. And down here in red is everything that has failed. Completely normal to see see a little bit of stuff that has failed in there. It is not normal if you see a lot of red versus no green. So that means that's a good indicator that you did not have a successful sequencing run. So these are just, quick little QC graphs you can take a look at just to make sure that everything sequenced and performed as you expected. All right. Next graph that we show on your report is your quality score. So this is going to give you your Q score for your samples. We want it to fall within this blue box. It's totally normal. There'll be a little bit of a spread, and that's to be expected. If you see this line here falling down here in the red, that's not good. That's an indicator that something went on with your sequencing run. So as long as this line is within these two bars, and it's hard to see on here, but this is like a blue color. As long as it's in there, you are good to go, because you really want to have a Q score of around 9. I think this one, yeah, this one's falling right within that range. All right, this is a very busy screen, and I realize the colors look completely different here than they do here, so I'll try to point this out to you a little bit. I don't expect you to memorize every single one of these colors and what each color means on here, but this is kind of giving you a breakdown of what the pore health is for each one of those wells that you have on here. The biggest one you really want to take a look at is, this is a nice bright green color on my computer versus this one. This is a dark green color here. So we really want to look at this light green here, which is basically, that's where your DNA is currently being sequenced. So that's good. You want that to happen. If this is very low, that's usually an indicator that something happened to your library prep. Maybe you washed too much of your library weight during the cleanup steps. But typically, this is what we want to see is this kind of good bar height in here. Again, apologize, kind of hard to see, but this one, I promise, is a darker green color. And these are just pores that are happy. They want to start sequencing. They're not sequencing, but they're there, so that's good. So we have a good number of pores that are available here. This yellow color here, where it says adapter, this is just basically where one of the pores in the channel has gotten another strand of DNA, and that's where that adapter is attaching on, so it's sequencing that first little adapter bit portion of it. So basically, comes in, sequences it, and then immediately moves over into this dark green portion of it. This color here is when it's unavailable, so that means that that pore has been blocked. With what we're sequencing, since we're doing amplicons and we're only doing 1 kb length in here, actually a little bit less than that, we shouldn't have to worry too much about the pores becoming blocked. Are you different? Happy to answer those questions later so we don't confuse folks. This darker purple color here, that means that the software can actually reverse the voltage on individual wells. So if a pore does become blocked, it will reverse the voltage to try to kick whatever's in there out so we can unblock it. And then that way we can hopefully remove that blockage and then be able to use that pore again for sequencing. And then these last couple ones where it's multiple saturated or 0, that typically means either you've got more than one pore available in there. Saturated, basically, if you see a big black bar, that's not a good sign. That's definitely an indication that something happened, usually during extraction and something didn't cleaned out of there. And then 0 just means either nothing's passing through that particular pore. Again, don't expect you to memorize all these. Again, we'll get you copies of this because I would be able to actually see the colors a little better on it. So it's good to monitor your health, and again, this is going to be the next little kind of graph afterwards. Every time we do those pore scans, this is letting you know how many pores do you actually have available, what's blocked, what's saturated in there. So like this is a good example where you've got lots of good happy pores. They're ready to start sequencing in here. And I realize this is probably very challenging to see, and I apologize, versus the next screen I'm going to show you. But But this is showing you an example of a really good Ampore run. So you've got good levels here of this lighter green color, so you've got plenty of stuff that's sequencing, and you still have lots of good pores available when you do your pore scans. Versus here, where this is a lower pore occupancy run, and I apologize because this is actually an example from like one of our RUO runs, because you normally would not see this much data collected on it. But essentially what they're trying to demonstrate here is you only have a little bit of lighter greens, that's just a little bit of your simple sequencing, versus you've got tons of pores here that want to start sequencing and are not able to do so. If you, going back to the bubbles, if you accidentally introduce a bubble into your run, that will affect your sequencing run, because again, bubbles cause your pores to pop out of the membrane. So that's going to ruin how much sequencing data that you get. So that's what essentially what this is showcasing here, where you've got a lot of empty zero pores starting out with in the very beginning part of the run. And that's a good indicator for us in tech support to let us know like something happened with that flow cell. And then lastly here, we're talking about, and if you have any type of channel blocking on here. Again, channel blocking typically happens if you've got something that carried over, usually from extraction, so that's going to cause those pores to become blocked and you're not able to start sequencing with that. And so basically, if you see something wonky like this, that's a good indicator that there's something weird going on with your run. All right, and then we've got the very last one here, just showing an example of if you have an osmotic imbalance of what oversaturation looks like, where you've got too much of this saturation showing up here. All right, we just have a couple more screens talking about the QC, and then we'll move over to the fun stuff of data analysis. We also show you the translocation speed. So that's essentially how fast your DNA is transferring through the pore. This should be around 400 base pairs per second, but we've got kind of like a little bit of wiggle room here, which I know is really challenging to see. But it's got to stay within, essentially, this. We've got two different bars that we can kind of let you know. And if it goes out of that, it's an indicator that something is going on with either the device or maybe the temperature in the room. Because we also monitor the temperature too. And that should stay pretty consistent. And at the very end of the run report, it's going to be the system messages, so that's going to let you know if there's any errors, any warnings. Anytime you've had a pore scan happen, it's going to let you know when that occurred on here, and then it lets you know how many pores were initially found and what's available for sequencing. So let me see what I can. Okay. So I get asked this question quite a bit. So you see where it says here, we've got over almost 1,400 pores that are available, and it talks about, or it found a total of 1,400 pores, but then 495 pores are available. So that's initially looking at every single channel, and remember, every channel's got 4 wells in it, so it's counting each one of those individual wells. And then it's breaking it down to how many of those channels actually are capable for sequencing. So that's the differentiation between those two different numbers on there. Because I've had customers call me freaking out. They're like, oh my God, it says it has this many pores. Why is only this number of pores actually sequencing? And I'm like, that's the reason why. You've got channels versus wells. And that's what it's breaking it down for. Alright, before I move into data analysis, is there any questions around the run report portion of this? Again, this is just trying to provide you with some QC information so that way you can get a head start of knowing whether or not this was a good sequencing run or not.
So my query is, you say the flow run will be valid for 60 minutes.
Pardon? Oh yeah, you do the initials, yeah, you've got 60 minutes.
If we want to run 5 5 sets in one group. So can it be coordinated accordingly? In one group we can 22 plus 22, like 88 samples can be done?
Yeah, if you've got it timed correctly and you don't have anybody interrupting your tech, they can probably get in and do it, where they'll be able to. This is where we probably have to sit down and have a conversation, like where does it make sense to do the flow cell check, and where in that process, so that way you can actually get that ready to go with all 5 flow cells at a time. But there's a way to do it. You just need to, we just have to look at the protocol and see if where to get each one of those libraries at, so that way you go bum, 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 bum.
Should all the 5 be loaded simultaneously?
I mean, you can. It just comes down to time management with your
Like when we have loaded one of the flow cell, while it's running, can I load?
Oh yeah, when you've got one going, yeah, you can go ahead and move to the next one, move to the next one, move to the next one, yeah. Because essentially you can go through and as you're getting, have your different preps going, as you're getting closer to like when you're doing the cleanup for all 5, then you can load all 5 flow cells on here, do the QC check, because once you finish up that cleanup, you're just adding your adapter, and that's just a 1-minute incubation with it. It doesn't take that long, or actually I think it's 5 minutes. It doesn't take that long to add on your adapter, add in your, by then, you'll be able, the flow cell check should be completed. You can go through and check, yep, all 5 positions are good, all 5 flow cells are good. I probably would already have my sample sheet already made up before I got to that step, so that way you're not wasting time creating the sample sheet portion of it. And then you can go through, set up a run, go through the run setup on there, load your library, that one can start, boom, move to number 2, add that library, go through the screen setup with that after library. So you can do that with it. It's just, got to make sure your technician is focused with the time management portion of it. Any other questions? All right, let's get to the fun part. Let's talk about data analysis. Okay, you've got a program in the background called Epitome. That is our bioinformatics software that we use. And you don't have to worry about directing the software over to do anything. The software is automatically going to move. What's this part? So, apologize, this slide's probably a little bit outdated since we got more additional information this morning. That there might be another catalog that has come out, but basically everything, all this is based off of the WHO 2nd edition catalog. And so these slides are based off of that information in there, so they're a little bit outdated since we are going to be introduced into the 3rd edition with it. But essentially what we're doing is we're working with that 2nd edition right now, which was released about a year ago. So this has all the updated SNP and indels in there, and everything that's been reclassified across those groups. And it also has specific expert rules that we're going to be including in here for those specific mutations. This is just a little bit more of a background because prior to this version of the software coming out, we had a beta version of the software. And so the beta version had a little bit different way it was calling each one of their genotypes that were in here. So this particular slide is just explaining a little bit about those changes in there. So previously, we were really only identifying the SNPs, some of those deletions, and any of those mutations that were classified underneath either WHO Group 1, WHO Group 2, and basically what we call an ONT call. And the ONT call really confused a lot of people, so we kind of changed that. And actually at the same time, the WHO catalog version number 2 came out, which was perfect timing. So we updated that to include both SNP and indels, so all that's being assessed in there. All of the variant classifications, those are all based off of the version 2 catalog. So some previously group 2 have now been reclassified as group 1. And so basically how we're looking at these various different mutations, we're breaking it down to either WHO mutation groups 1 and 2, or WHO expert rules, and any type of full gene dropouts. So here, it's just basically broken down by the groups, what their endpoints are, and basically it's a little bit of notes about it. So obviously with group 1, very strict criteria. We know those are heavily associated with resistance. So with group 2, we know it's a little bit slightly relaxed from group 1. Maybe this will move to group 2 will move to group 1. It just depends on more evidence that we collect. Export rules, so this is going to be any type of calls based on some type of combined resistance. So maybe something is in group 2, with the way the different mutations are combined, we expect that to then compute resistance. So that's why we have this export rule. And then lastly, if we detect any type of deletions, that will be in here too. So this is essentially what the algorithm looks like with our software. So we look at the coverage information. Do we have enough coverage for that particular area? Is there any type of gene dropout? Yes. Does that cause any type of loss of function? If it does, we're going to report that out. Now we're going to look over at this part where we're looking at our SNPs and indels, and we're also looking if there's any type of sort of large indels. We're going to go through and phase that information. So basically what phasing is, we're going to line up each individual read. That's what we call phasing with it. And I'll show you an example on the next one. And then, because we want to look at each individual read and see, is there a SNP or an indel on that individual read or strand of DNA that we sequenced? And we need to line all of that up and see, is that a real SNP? Is that a real deletion? So we can look at that. We're going to annotate that information, and then we're going to move forward. Is this showing up in group 1 or group 2? Is it in that catalog? If the answer is no, then we move down to, well, is this part of that expert rule that we're talking about? If the answer is still no, then we're going to discard it. It just might be just some random mutation at this point in time that's just of no importance to us. If going back up here, if it is in that catalog, then we're going to report that out. And then coming down to this expert rule, if it falls under that expert rule, then we're going to report that out as well. So this is just kind of like how the algorithm works as it looks through the various different mutations that have been discovered. So this is an example of what I was talking about earlier of phasing. So we took each individual strand of DNA that we sequenced and we're lining them all up. We're not just kind of combining it together, giving you an average and saying, yep, that's it. No, we want to look at each individual one. We really want to know, is this really a deletion? Is there really a SNP detected? So that's what this example here is showing, that we can confirm that this is an indel with this particular sample here. So this is one thing that kind of makes it better when you're working with the long reads. Are we able to detect these larger deletions? And the answer is yes. We are able to do so. So with this particular example that affects the PNCA gene, PIC gene, we're able to detect if there's a large deletion there or not, because that is going to affect, here, I have to pronounce, pardon me if I pronounce this and butcher this wrong, but the pyrazine resistance in here or not. So basically, we are able to detect this very, very large deletion because we have the longer reads and we covered that. And essentially what we're able to demonstrate is if there is a dropout of this particular amplicon, what we do is we go through our software algorithm here. You know, does the rest of the sample have very high coverage for the other genes that we're looking at, the other amplicons that we're looking at? Okay, the answer is yes. You know, do you have more than 10x coverage? So basically that means you have more than 10 reads that are covering that area. And if we want to make sure, does the positive control pass as well? Because if the positive control didn't pass, then that's probably an indication that probably the PCR was not very good. We might need to repeat this. But if the positive control does pass, then this is a good indication that we have a very large deletion event occurring, and therefore we can accurately predict this and call this correctly with it. So they did a proof of concept here, showcasing that we were able to accurately call this large deletion, whereas when you're working with Illumina, because they're such short reads, they were not able to cover that area, or not, but because we have, let's go back up here for a second. I'll just pretend that this is a very large because we have such long reads, we can cover that span of it as well. Okay. So this is breaking down the bioinformatics side a little bit more of it. So when we start looking at SNPs, we are able to detect down to, we can actually go a little bit lower, but we put the cutoff at 15% because this is where we know 100% of the time we're very accurately able to detect this. It can go a little bit lower, but that's where you start to lose a little bit of that confidence with it. So that's why we have the threshold set at 15%. So we have this set at 15% allele frequency. So essentially how this works is, in order for us to accurately say, yes, there is a SNP in this location, it must be presented in at least 15% of those reads. So basically at the frequency here of 0.25. This is where it kind of gets a little bit confusing with the algorithm, but essentially what it means also, it must be supported by at least 5 of those reads that we collected in that area must contain this SNP in it. If there's low coverage or some sort of imbalance, the software defaults to a different type of allele frequency. It actually changes it to a 0.4 because we really want to make sure that we're accurately calling this correctly. So essentially it gives you a stricter criteria with it. And moving down to our controls and different thresholds that we have. So with our controls, we're saying that you need to have a minimum of 20 reads, or again, 20 strands of DNA that have been collected in that area for us to be able to call that a sufficient area of coverage in there. Ideally, if you want to be a little more accurate, we'd like to have at least 40, but we like to say at least 20 reads are covering that area. For our NTC, basically our negative control, it will fail if 3 or more of those amplicons have 20 plus reads each. So that's typically an indicator of either A, you've got a contamination event happening, or maybe you mislabeled your samples on your sample sheet. But ideally, you should not see anything being amplified in your NTC. For your positive control, you need to have at least a minimum of 22 out of those 24 amplicons. They must have at least 20 plus reads each, and the NTC should have passed as well. So basically only the NTC should have the internal. I have examples of this too, so this will make a little bit more sense as we get into it. Because it took me a minute, I was reading through this when I was first learning this algorithm. But basically it's just kind of giving the background information of it. And then when we move forward to our patient samples, it will fail if it has less than 20 reads, so basically less than 20 strands of DNA that are covered in areas. That amplicon will fail. So when we were talking about earlier about having those samples that were right at that LOD level with it, and where you might get some of the amplicons showing up, that's essentially what this next little piece of it is. So if we have 10 out of those 20 amplicons or more, they generate 20 plus reads in one sample, we're going to say that that particular area has passed, and then we're going to give you at least a little bit of the drug resistance information for that sample. We're not going to be able to give you the full picture of it, but at least we can kind of give you a general guidance of maybe which direction you can go with it. So samples that typically are like this again are those that are either right at that threshold of the LOD, or they might even be considered negative in it. And again, we also want to take a look at to see, was there any amplification with the HSP65 too? We also want to look at our QC controls as well. And this is all locked in stone. We cannot make any changes to the algorithm on this. All right, so again, with reporting, you're going to get your run reports, your run report on this side. This is going to give you like the full printout of every single sample on there and just a quick little snapshot of what drug resistance are available for it. Then we can actually click on each individual sample, and then that's going to give you a little bit more information with it. So you can actually look and see what's passed on here with what are the quality checks here at the top and passed. And then, again, I apologize, it's hard to see with the colors on here, but you can also take a look to see what's resistant versus what's not resistant. So it's going to give you your run report again, it's going to give you like that longer page with everything listed on it, and then your sample report is everything that's going to be kind of like a one-pager on there. This again, this is all based off of our published guidelines. So this is kind of just giving you a little bit of background information, and it may or may not be on your crossword puzzle. These are the type of files that are generated on here. So our base call files are called FASTQ files. So each one of your folders are going to include everything that's barcoded in here and include all of your FASTQ files in here. This is just for your background knowledge. You don't have to mess with any of this. The software is going to do this for you automatically. So essentially, all your base call FASTQ files are generated, and then what it does is it breaks it apart into the separate folders, and then the software is going to take each one of these folders with your past reads in it, and then move it over to your analysis portion. So as a reminder from previously, we want to make sure that our sample sheet was set up correctly in the beginning, because it's going to also take this additional information into your analysis, because if it's not correct, there's a potential that your run may fail just from a simple error of you didn't label positive control in the right spot or etc. like that. So that's why it's very important to make sure that we have everything labeled correctly. And so you don't have to worry about the file format, because all of this is already going to be pre-populated onto the device. This is just a little bit more background info. All right, let's get into a little bit more about these analysis reports on here. So as a reminder, this is what the main screen looks like for your analysis report. So it's just going to show all of the various different samples that you have on here. Anything that's red is those are the drugs that are drug resistant. Anything that's blue, they're not resistant to those. And it's a little bit hard to see, but yeah, it gives you a little bit more information here. And unfortunately, if I try to blow this up, it's going to get even blurry, because it's already a pretty blurry screen. So the first thing you want to do once you've completed your run and you're looking at your completed and analyzed data is you want to look at your controls quickly. So basically, did they pass or did they fail? So if everything passed, you're going to see everything green, and then basically this is going to break it down to, oops. It's going to break it down by your negative control versus your positive control. So essentially this is showing which one of those 24 amplicons are those genes that we put together, which ones amplified, which ones did not amplify, and it kind of gives you an idea of how many reads that were collected with it. So it's kind of hard to see on the scale here, but for example, down here, this is your positive control, and here it got everything well over 20 plus reads for this. Here's your negative control, and then really the only thing that's amplified here is just 1. So as a reminder, you need to have 3 or less for your negative control to pass. And then over here is a good example of your run failing. So over here, on your negative control, we have 3 or more areas that are amplifying, so that's bad, so that's automatically going to fail your MTC. And then down here at your positive control, this is not good. So you can see a lot of these, nothing amplified, or these are way low, 20 reads on here. And again, it's a little bit hard to see on this screen with this, because it's hard to see the scale that's on here, but this is an example of something that has failed on here. Going back here to your negative control, this is possibly like a contamination event, so that's why it's really important to make sure that we're cleaning our benches and everything and have our pre and our post PCR area. Okay, if we come down to our sample section, and again, this is when we're looking at that big clean report with it. This is going to give you kind of just a quick overview of the sample. It's going to give you that species identification. It's also going to list the oligotyping on there if it is TB, and then give you the drug resistance again. We only do the drug resistance for TB on here. Basically, samples once again pass if they've gotten enough of that Ampore TB amplicon coverage. Do we get that HP65 coverage? And then did that internal control pass or fail? So as a reminder, the internal control is a competitive control, so if it's a positive TB sample, that internal control should not be any type of amplification in there. So for your drug resistance, again, all the various different antibiotics here are listed, so the color is going to dictate whether or not there's any type of resistance mutation detected or not for that particular amplicon. Red means that it's resistant. Blue means that, like this bluer color here, means that there is no resistance detected. And then orange, if you see orange, that typically means that it's undetermined. And that usually happens if it's like a sample that either the PCR didn't work for or it's on the lower LOD side. There's just not enough coverage in there to determine whether or not there is a type of resistance with it. But for this like particular patient, we know we can take this drug right here. They're okay to take that one. If the sample fails to amplify any of the TB target, but amplifies the HSP65 target, the sample will pass and the NTM will specify only for that particular sample. And then, which is basically, I think the last one that's showing. And then if the IC fails, then of course the sample will fail. That's your internal control is basically making sure that everything is amplified correctly. If we go into our sample detail view, this is going to give us a little bit more information on here. So up here at the top, this is going to give us all of our QC information. We've got here our drug resistance information on here. It's going to let us know, again, if our internal control, everything passed on here. Again, you want to make sure we have at least 20 reads minimum for each coverage. And it does list a variant table down here at the bottom, so it's going to tell you which drug-resistant mutations were detected, the gene position, the protein change in there, the allele frequency, and which one of these mutation groups did this fall under. Why did we end up calling this as a resistance in there? Going down on the report, it will actually give you the gene coverage on here. So this one is actually a really good example of a very large deletion right here. But we can see that the internal control should not amplify because, again, this is a positive sample. We should have our HSP65 on here because that's going to give us our species identification. We should have at least 20 minimum reads on here. And again, as a reminder, for our sample, it only will pass if you've got 10 or more these amplicons here that have been successfully had enough reads for them. And this excludes the QC and the HSP65. If it's less than 10, then we're going to end up classifying this as being a failure on here, but it might still pass the HSP65 and the IC criteria with that as well. So going back to previously where I was talking about that 15% allele frequency. So again, this condition will move to this point for allele frequency default. Again, this is just a stricter criteria that we utilize for some of these samples. This only defaults to this if the sample has less than 22 amplicons, so it needs to be in that 10 to 20 amplicon range, and the sample coverage is much lower. So that occurs, then we're going to move to this higher allele frequency because we really want to make sure we are accurately calling these resistance markers. more reads. Ideally, you should have all 24 in there. Samples with an amplicon count between 10 and 24. There might be some areas where it's undetermined. So basically, if we got a lack of coverage where this is showing right here in red, that means that where we just can't accurately say yes or no, are they resistant to this particular drug on here. So for this particular example, we are unsure if they have a thin is that how you pronounce it? Resistance because we are missing this Byramjee application here. It did not get a full 20 reads. They got a couple of reads, but not up to 20. So therefore, the sample is going to fail on here. And any sample, again, which fails coverage, where it has 9 amplicons or less than 20 reads, if we're not going to get any type of drug resistance report, because again, we don't want to accidentally call something out correctly. I'll just take some water. Quality of my voice is going.
So take a 5 to 10 minutes break, so that we will get some break. 5 to 10 minutes, we are still continuously speaking. In the meantime, you can solve the crossword puzzle. He said he has already got 9. I think you also got one. One shot. One shot. Okay, you have also
it's only going to amplify and go through analysis when the other targets do not amplify on there. So this is used to detect if there's any type of faults or failures within your PCR amplification through library prep or sequencing of the sample. So essentially what we're showing here is where we've got no amplification on the IC, but we do get amplification of the TB targets. That's totally expected on here, so we do pass this sample. So that's what's showcasing right here. We got 0 amplification of the IC, but we got plenty of coverage here on the rest of these amplicons. Versus over here, where you can see we've got a lot of stuff here that unfortunately did not amplify or did not amplify well. And then we still look at the coverage here for our internal control of this one. So we will pass this sample, I should say pass it for the internal control, but we fail the rest of the sample because we just don't have enough coverage that's on here. this core sample completely failed. So we can look and we see we've got no internal control at all that amplified. So therefore, that's a good indication that this core sample needs to be repeated. And I do have an example that I can show you of that one next. So essentially, this is kind of like the bioinformatics workflow, sample QC with it. So we do our first initial run assessment. So we look at our negative control over on this side, and we're going to assess that control. Did it fail? Then, I'm sorry, the complete assay failed. If it passed, and then we also look at your positive control too, did this pass or did it fail? If either one of these fails, you get assay failure, and then we also look to see did they pass? Great. Now we can move forward into the assessment of our samples. So then we look at our test samples here. We're going to assess the sample coverage. Did we get more than 20x reads? Great. Then we're going to move forward into saying that sample passes. If that sample coverage fails, we move down to our next criteria of looking at that internal control. Did the internal control pass or fail? And depending upon that, we will either pass or fail that sample. And so this lastly is the single sample report that we have on here. So this is typically kind of like what you would pass over to your clinician. So that way they can see what's resistance versus what is not. It's just much easier, simpler. They don't have to review all that rest of that data. Unless that's something you're interested in, they can pass that along to them. But typically this is where you just kind of want to give them. Just be given that sample coverage on there and the table of what antibiotics are a go or no go on here. We're not going to give you the information of sample coverage or any of that other information. Because it just starts to get a little bit easy for that. Alright, so before we move to Q&A, I'm going to actually show you guys some real numbers. And I'm sorry, I have lots of tabs when I was working on things. Okay, so this is run on a little bit older version. So we're not going to have this Pliego typing information on there, but that's okay.
I have a question. Now with these mutations, we are taking sputum as sample, right?
Yeah.
It's not a single or pure colony or pure strain.
No, we're not spraying from sputum.
Yes.
Yeah.
So there is a possibility that we have multiple hetero infection.
It is possible. Yeah.
How do we resolve that kind of thing?
So it should, well, you guys can kind of take see like what's the mixture, I think, of that culture. I'm just thinking through it. Yes, it should get the resistance information that's in there. I can probably see there might be a case area where we the resistance TB that you have in there versus the non. Lower. That's a good question. I'll take that back to the team folks and see what the answer is for that one here. Yeah.
These algorithms is like to set for a lower mutations. If some mutations, the algorithm for bioinformatics analysis. So there is a set algorithm for the lower mutations. And that new mutation is come, so we can get the idea. We can go for another.
So you're talking about this mutation. So anytime that the
these mutations are the more mutations.
Yeah, these are the ones that have been approved by the WHO. Yeah, so when the WHO does update their guidelines, we'll be updating the software as well to reflect that. Yeah.
The lower mutation is there. There is a mutation they are not given in the list, but some is a new, it is not matching unidentified mutation, then go for the sequencing analysis.
Yeah, reflex to it. Okay. So at the top part of this report, it's going to tell us the total number of samples that were run, how many samples passed versus how many samples failed. Again, we have our run status up here. Did they pass? Did they fail? If you click on View Details, that's what's going to populate this screen. And so it's going to give you some points of note at the top exactly how a run failure occurs. with this and maybe how much the specific amount of coverage that you need to have. So looking at our no template control, again, we need to be 3 or less amplicons with, if 3 or more amplicons are covered with the medium coverage of 20x, then that sample fails. So we can see here in this particular example, yeah, we've got a few random reads that came through, but we're only looking at like 1 or 2 reads. So we're way below the 20x coverage. That's why we consider this to still be a passing passing negative control on here. And then down here we can assess our positive control. So with the positive control, again, we want to make sure all 22 amplicons are covered. And then you have a medium coverage of 20x or up for each one of these amplicons to pass. So we can just get it bigger. Okay. So here you can see where we've got our internal control here. Again, because this is the positive control, therefore this is a competitive internal control. It did not amplify, so that failed. That's perfectly fine. And then if we look here, we're way over the 20x coverage. So that's perfect. So that means that this particular positive control passed. So here are some examples of the different samples that were run. So we can see here, what was the sample ID name, what was the barcode associated with it. And this might look a little bit different than the report that's going to be on this, because again, this is a report that was from the beta testing, but essentially it's going to be very similar on here. Then we're going to get the information for here, is this TB, yes or no, and what is the lineage for it. Then we're going to get our first and second line drugs, and then what is resistant versus what is not resistant. So I'm just going to kind of scroll through this here so you can see we've got different types. We've got different types of lineages that have been detected with these samples. You can see we've got various different types of what's resistant versus what is not. I'm going to actually click on view details for one of our samples. So for this particular sample, you can see how many reads were actually collected for that one. And then this is going to give you that quick information here about whether or not that sample passed. So we need to look at the sample coverage pass. Did we get that HSP 65 coverage? And did we get that internal control coverage? So if you click on details, you'll get this pop-up on here. And I'll give you again just a little bit more background, points of note on here and what we're looking at. So again, for coverage, you want to make sure that we're having at least 10 amplicons with more than 20 reads in order to consider that as a pass. So these are all the different targets that we're looking at. So it's not going to show it all on one screen, so that's where you can actually scroll through and look at the different ones and look at that different information there. Then we've got our HSP 65, and then our QC. So again, as a reminder, the QC should have failed because this was a positive sample. Okay, we have our resistance summary here of what's resistant versus what's not resistant. These are the variants that was detected for this particular sample. So you can see the position, reference versus what was found, the detected frequency for it, etc., and then what category this fell under. So this particular one was underneath group 2. Okay, we've got our coverage. So again, QC failed, and that's totally expected. And then you can see how much coverage that we've gotten for it, which is well over. So you can see this is well over the 20 reads, but that's excellent for this particular sample. Here is the text taxonomic information for it. So as a reminder, it's going to list all the species that was found with it, depending on how many reads it was found for each one of these. So say, for example, it found just one read for this particular species right here. The majority of the species is still TB. And again, this is an older software version, so that's why we're not showing this oligotyping on here. Okay, this is an example where we ran 22 samples and then 2 of them failed. And for these particular samples that failed, it just came down to the extraction method that this particular lab was doing. So there's just a lot of PCR inhibitory reagents or factors that were in it that got kind of carried over, so that's why this failed. So if you look at this, it just ended up killing the internal control, and that's why that failed. Because again, it's going to look at, okay, you know, did this fail, did this fail, this failed, so all of that failed, so nothing amplified with this. And again, we can see that we're not getting anything with it. So something came along for the ride and completely killed that PCR reaction with it. We just got a little bit of coverage for this one, but again, based off the criteria, because we have no internal control, we failed this one, because this internal control only came in at, let's see, yeah, not even 20 reads. It was just shy. It was probably about 15, 16, about 17 reads we got for this one, so that's why this particular sample failed. What we don't want to see, when you run 22 samples, all 22 samples fail. So what do we think happened with this particular run? Any guesses? Okay, anything else? So this particular run, they actually messed up their sample sheets. So they flip-flopped it. Well, I shouldn't say they messed up the sample sheet. They flip-flopped the PCR barcoding plates. So they made their sample sheet correctly, but instead of having this as position A at the top, they turned the plate and used these barcodes, and so everything was completely backwards. And so because when they did that, positive control, negative control were mislabeled on the sample sheet, so therefore their run failed for that one, yeah. So how we were able to troubleshoot this is when we looked back at the run report, and we could see they got tons of reads. They got reads there, so therefore they should have been able to do that, and we realized they had to go back and look at the plate and realize they had completely flipped the plate the wrong direction. So that's so yeah, so did you see everything failed on here? So what do we do with the data that we get? Because this was the RUO version, we were able to manipulate it. With this version, this is going to be locked down, unfortunately, so we're not able to do that. Because that was our secondary test. We tested out and see if we just redo the sample sheet, run through analysis again, and that's how we definitely confirmed our suspicions with that one, and also looking at the plate, and they oh no, the plate was flip-flopped the wrong direction. Yeah. Your guys are not going to do that. We're always going to make sure that A is at the top. Yeah, it was one of those things where you just make that mistake once, and you just never do it again. Let's see, if there's anything else fun in here. Yeah, so here's one where you can see we've got kind of like this orangey color, so let's take a look at this guy. Oh, that's because everything else was blue, or seemed to be, see here with this particular target, where we did not reach 20 reads. So this is an example of, we had over 10 amplicons amplified, over 20 reads, but because this one particular target here did not fully amplify and give us the full 20x coverage, yeah, we got about less than 5 reads for it. That's why we're getting this orange color. So obviously there's something within that gene that mutations in there dictate whether or not you can be able to take this drug or not. But this patient has got plenty of other options that they can look at and they can take this one. And then we have our nice sequencing summary at the bottom. So basically it's just going to list all of your samples on here and kind of show you all the full coverage that we see. So you've got tons of coverage and samples, or reads that would look like for each one of these samples. Okay, any questions on the rep report, reporting?
Can we give the lineage in this?
Oh, I'm sorry.
Can we give the lineage information when it talks taxonomic classification?
Yeah, I'll pull it back up. Well, oh no, unfortunately on this one, it's going to list it, but because this is an older software version, we're not able to see that Spligo, like the nice pretty graph that's on here, but it does list it on the report here. So you can see right here's your lineage. So you've got your base shading for this particular sample that they've had here. This particular one had a couple of different lineages with it. This one did not call anything because it's below the threshold. So you can see that basically it did not get those 150x coverage with that, so that's not on there. Unfortunately, because there's a bug in this particular version of the data analysis software, I can't troubleshoot a little bit further and see how many, what the coverage it was for it, but that's why. Which will let you know yes or no with that. Any other questions? Yes, you'll be able to transfer that raw data off. So you'll get your raw data, those FAST2 files. You'll be able to transfer that out. Because, for example, let's say that you run this test and 6 months later or years later, the WHO catalog comes back and changes something for it. You want to be able to then go back to that raw data. I don't know if you're going to have the ability to reanalyze it yet within the software. That's something I can take back to R&D, because that would be lovely to reuse the same software to point clicking before. So I'll ask them that question for you, because it's something we have to think about anyways. But at least you save those FAST files. That base call data will be in there, and you have the ability to reanalyze that in case there's new mutations and things of that nature. It's also good for like validation side of their software changes. Yeah, those are the FAST files. So that's the raw pixel data. It may or may not, because again, this version is a little bit different. This is an older version. Yeah, so right there, these are the mutations that are on here that therefore are saying that your resistance to these drugs here. So this is the reason why. Yeah, and that affects these type of drugs on here.
How does it tag the novel mutations? Novel mutations, new mutations?
Yes, that's where it's not going to tag it in this report because we're only looking at the class 1 and 2, so that's where you have to go back and look at the raw data with it. They're not going to list mutations that are not within that group 1 and that group 2. No, because based off of how this algorithm is written, we're only looking at group 1 and group 2. So if you have a brand new mutation and it's not in group 1 or group 2, it's not going to list it on this report. So that's where you have to go back and look at right.
Even if it's not matching the wild type,
no, it's not going to do that. Because if the WHO has not determined that that mutation is of importance, it's all going back to the WHO with that. So that's where they're basing the algorithm off of it. Yeah, I used to support HIV, drug resistance mutation, an assay, sequencing assay with that. They would list every single. They would put at the top like these are the ones that reason why you can't take a certain drug, but they would also list all the other mutations in there. But unfortunately, we don't do that with this one. That's additional feedback I can provide because it'd be nice to know too. Yeah. But right now, yeah, they're just focusing on group 1 and group 2. Unless the expert will. Any other questions? Yeah.
In the event that you get some very unusual mutations, by unusual, what I mean is amongst these submitted ones, say the newer second-line drugs, is there any kind of protocol that requires us to repeat or reflex or confirm this phenotypic? Is there anything at this moment?
Not at this time, so unless the if the guidelines get updated, then we will update the software to reflect that because we want to be with the most current version of it. But at this point in time, no. It all makes sense. You guys got it? Good drop, run. So I'm going to, with my colleagues, I'm going to convert this presentation that I gave today into a PDF, so that way you all will have a copy of it and be able to access it, especially like screenshots from like reports and like that.
Then you close this and then switch on the start. But all of this happens over here. Okay. But if you're comfortable, you can also do document tables like over here. I generally encourage people to do all of the research in the machine itself.
So that's what we call just like
the only reason why I don't recommend it is sometimes you can actually, if you drop into something like that, then so these are expensive, so you pick one.
So it was 1.5 or 1.9?
Those people you should just get your go ask the other one there. It will be black like this. If it's empty, it's going to have a gray. Because if you see, yeah, I mean, if you can basically just see the side of this is big.
So after this thing, we need to add dedicated micrometer of priming solution. In addition to the priming solution in a micrometer, then again they are 200 to review micrometer of plastic.
Put it in the machine, no, I mean, see, in academics, they will use it and use it and use it and use it. They will, it's like there's nothing to it. So I guess this they will
No, is that because the reagent what you
I think she mentioned there is a DNA or anything barcodes that are left over here. So that's done twice. So it's like a 5-minute incubation. So you do one, 200, wait for 5 minutes, and add another 200. Yeah, you can do whichever. I mean, some people prefer yours, some I prefer this.
With you. So we thank you very much for attending this particular workshop. Now we'll have a group photograph. So would we assemble somewhere right now?
We can assemble outside.
Come to
Hello, I met you. Yeah, we met you. Yeah, we met you.
One second. Yes.